So, welcome everyone to our second hand-on graph in the brain. Today is going to be a shorter version of the previous one, but we have 1 day of workshop. And just one point that I have to, one reminder that I have to give you. If you have questions or comments, we ask you to do it in the microphones because we have people watching on YouTube. So in order them to follow and have questions and send you questions also, okay? So we will start with André Fujita, which is our new NeuroMat member, and he's gonna present our first seminar.
Okay, thank you very much. Well, it's my great pleasure to present and to introduce Professor Peter Stadler from Leipzig University in Germany. Well, as you can see here, he has many affiliations. He's a worldwide professor. And he's my colleague. We are working together since 2017, right? 2017. And the presentation here is about autocatalysis and unstable cores in parameter-rich chemical reaction networks. Thank you very much, Professor Stadler.
Thank you, André, for the invitation. So I'm here as part of an exchange project that we have had going on for a while. So it's a pleasure to be here again and to present a little bit of my work. I realized that I will speak about exactly nothing that appears in the title of the conference. So there will be no brains. Instead, I will speak about metabolism mostly. I will not speak about random graphs, but instead about hypergraphs. But there is some randomness in a way in there, since I basically look at randomized reaction rates. So I will talk about structural features that are independent of the detailed choice of the parameter. So let's see if I can get this. Almost. So what's... it did something. Oops. Okay. So our object of discussion today is this kind of complicated networks. So this is a depiction of a very small part of the carbohydrate metabolic. What you see here is all the big blobs are chemicals. So those are the vertices in our network, and what you describe in a metabolic network or in a chemical network in general is the transformation of chemicals through chemical reactions. And the chemical reactions are given by this other type of vertices here. So what I'm using here is basically the so-called Koenig representation or bipartite graph representation of a hypergraph. So the little squares essentially represent the hyperedges. My graphs or hypergraphs are all directed. So every chemical reaction has a direction. It goes from some set of reactants that react with each other, as the name says, and produces a product. And so what this reaction basically, or what this little network essentially is just supposed to depict is kind of the structure of the things that we are looking at. For today, we will actually not look into the inside of these blobs, so we will not care about what the individual reactants actually are. In this case, this is just different types of sugars, but we will only be interested in what's called the stoichiometry of this type of networks. So the way how they, this is the right direction. So what we will be interested on is if the whether there is anything that useful that we can say about the dynamics of such a network, given that we only know the underlying topologies, that we only know the connections in this network and nothing else. So no specific values of parameters and no details otherwise. So what we want to know is, can we have conditions for there being stable fixed points? Can we look at the existence of oscillatory behavior in these kind of networks? Can we find out whether particular setups or particular structures of these networks allow for bistability or multistability or not? And the way how we are going to do this, I will outline in a moment. We will need a little bit of notation to understand what's going on here. So a chemical reaction basically consists of a bunch of molecules. They will always be called something like M, from M1 to MM. And they come with coefficient, which is just multiplicity. So say 1 M1 plus 1 M2 plus 3 M3 will react to 2 M4 or something like this. So this is how you read this. These stoichiometric coefficients for educts or reactants are written as S, and the lower index is the molecules. The upper index is the refers to the particular reaction that we're looking at, right? So if you look back to our messy network here, and probably I will make this wrong the whole time. This messy network here, there is lots of different reactions in there, and this index J just runs over all these networks. We make one assumption that is not really a crucial assumption for the whole theory. It just makes life a lot easier to assume this, is we assume that there is no direct catalysis. So one of the coefficients for a particular molecule, either on the educt on the reactant side or on the product side, is 0. So we never have an explicit catalyst. So we never have 2A gives 3A directly in one reaction. If we want to model something like that, we can always just introduce an intermediate complex, whatever this catalyst is doing, forming some intermediate molecule, say an A dimer or whatever, and then we can just pull apart this reaction into 2 steps and get a network that has the same net effect, but again doesn't have this explicit catalysis. Under the reason why we want to have this assumption of no direct catalysis is that under this assumption, the whole structure of the network is given by the so-called stoichiometric matrix, which is simply the coefficients with which a molecule is built minus the coefficients with which it is consumed. And from that, I can just, under this assumption, reconstruct the whole graph so that you can think of this stoichiometric matrix just as an incidence matrix of that directed hypergraph. And what we want, therefore, is we want basically to have statements on this incidence matrix S that links the structure of this incidence matrix S with dynamical properties of our model. So if we talk about dynamics, so let me just briefly go into what the dynamical system looks like that we want to be working here. We have for every molecular species, we have some concentrations which necessarily are non-negative numbers. So it's kind of difficult to have -2 atoms of -2 molecules of something. So this is obvious, and we will also assume for simplicity of the math that we can do everything just in terms of differential equations. So we will assume that we have concentrations that are real valued, and we can write our dynamics as x dot, so that the temporal derivative of the concentrations is given by the stoichiometric matrix times reaction rates for every reaction. So this R now is a vector of reaction rates indexed by the different reactions, and the reaction rates, of course, depend on the concentrations of all the stuff that is swimming around in our system. So, wrong direction again. So just to have a look at this stoichiometric matrix again, what this stoichiometric matrix really encapsulates for every reaction, which comes as the column. It basically says, okay, so 2A plus B gives C plus 3D. So we consume 2 A's and 1 B and produce 1 D and 1 E, right? That's just the way how you read this thing. So it's really just the incidence matrix. And what we want to talk about, as I said, is the dynamics of this model under the assumption that we don't really know a lot of details, or we know next to nothing about the reaction rates R. This sounds like a rather hopeless program in a way. So of course we have to make some assumptions on our reaction rates. The assumptions that I'm making are very, very weak. So I will only assume what's called a monotonic kinetics, or monotone chemical functions, which says, okay, so reaction rates are always non-negative. So if I have a reaction, it always only goes forward, or it doesn't occur at all. That's my first axiom here. The second axiom says that if I have a positive reaction rate, well, then all the products of that reaction have to be present in non-zero concentrations. So the reactance of a reaction has to be positive for everything that goes as a reactant into my reaction. What I also need is that, so remember, those are the coefficients on the reactant side, right? Not the ones in the stoichiometric matrix. This is minus the ones in the stoichiometric matrix. And what I also want is that if a molecular species, if a molecule does not take part in reaction J, then the rate of reaction J does not depend on its concentration. So we basically assume that all interactions in that system are described by chemical reactions. So you can't have things that you say a molecule that on the side binds to some other molecule and shields that, so effectively reduces that. If you want to model that, you basically have to model this binding reaction explicitly as a chemical reaction and throw it into the system. So there is no kind of unaccounted for weird effects where something that is not part of a reaction affects the rate of the reaction. So these the final axiom here says that if I take a reaction, I have a reactant that is part of that reaction. If I increase its concentration, that will increase the rate of that reaction. In principle, so it's convenient to have this the strong inequality here. One can largely get away with also relaxing that to an larger than or equal, but we don't really care about this in the moment. So classical dynamic system theory, just to remind you, the stability of any fixed point in such a system is given by its Jacobian matrix, which is simply the partial derivatives of this of my vector field. And what I want is I can also look at the partial derivatives of these reaction rate functions and get a matrix that I want to write in this way. RJM is the partial derivatives of the reaction rate of the jth reaction by the mth molecule. Remember that if M is not the reactant of reaction J, then this thing is identical to 0. So we have a bunch of 0 entries, and we have a bunch of entries in this R matrix that is strictly positive, right? So RJM is always positive if M is a reactant of reaction J, and it's zero if it's not a reactant of reaction J. A short computation gives you that you can write the Jacobian of our dynamically system at some fixed point simply as G of x is S times R of x, where so now S is the stoichiometric matrix as before, and this R of x is a matrix that kind of recapitulates all the negative entries in the stoichiometric matrix, right? Because just with basically arbitrary real values. So what we are now interested in is what can we say about the Jacobian given that we only know S and this very crude structure of R, but no numerical values of the R. And so in order to formalize this, we don't know anything about the R really. We introduced this notion of a parameter-rich kinetic model. So in general, if you write down a chemical reaction system, then these rate functions, R of x, will depend on a bunch of parameters. Individual rate constants, dissociation constant of intermediate complexes, or whatever you have that's kind of hidden from the rest of the system and doesn't play a role, temperature, pH value, what have you, right? So lots of different values that can go in there. And so what we are now interested in is, can we write kinetic equations that have enough flexibility depending on the parameters that we can do the following. What we want to do is we would like to have, whenever we have a positive equilibrium, so a concentration vector with all positive entries such that S times R of x is 0, so that's an equilibrium point in our dynamical system. What we want is that for every choice of such an equilibrium point, we can, first of all, ensure that these coordinates of x are fixed, and we can choose entries in this matrix R. We can choose actual parameters for these reaction rates, so for these partial derivatives, such that whenever this RJM is positive, that RJM is positive if and only if SJM is positive. So exactly for the reactants in reaction J, we always have a positive entry, and otherwise we have a 0 entry. And what I want is that there is a choice of parameters that depends on the matrix that I want to have and on the coordinate, the fixed point that I want to have, such that we have these partial derivatives that we can choose, and we can choose the fixed point independently. So we have enough parameters to pick the fixed point and the numerical values, not the signs, but the numerical values in this matrix R. So now this sounds like a very, very strong condition on the type of kinetic models that we want to have. The surprising result, or the surprising observation, is that this is actually something that's, as I will show you in a second. Maybe let me show you this slide first. So standard models of reaction kinetic actually have this property of being parameter rich. So in particular, already Michaelis-Menten kinetics, so enzyme catalyzed reactions, have enough parameters, enough degrees of freedom, just by the fact that the reaction works through a binding with that enzyme that otherwise is not part of the chemical reactions, to give you a parameter-rich system. There is another type of systems called generalized mass action kinetics, where instead of the stoichiometric coefficient, you have here essentially an arbitrary non-negative exponent up there that is also sufficient to parameter. The proof of this is very boring and just goes by direct computation. So you, in both cases, you can just invert equations and just calculate what the parameters have to be for the fixed point coordinates and the partial derivatives that you want to have. Unfortunately, this is not true for mass action kinetics. So in the case where this exponent here is given by Sijm, then you don't have enough parameters that is independent of each other. So the results we're talking about do not work strictly for mass action kinetics, but at least they give you an indication of which architectures or structures one might want to look at. So why do we care about these? So what we can do in these parameter-rich models, we can fix the coordinates of the equilibrium, we can fix the values of R. So what we basically can do is we can ask, given S, is there a choice for this R that we can construct with these constraints on its sign, so that, for instance, we get an unstable fixed point or we get a Hopf bifurcation. So we can basically ask, can we take the structure of S, right? So does the structure of network admit a certain interesting type of dynamical behavior, at least if we tweak the parameters? So what that basically means is there has to be a choice of R such that S times R has, for instance, a positive eigenvalue, or there is a parametrization of R that takes you through a Hopf bifurcation and so on and so forth. So this is what we want to do here. So the goal is basically to say, okay, if we have a parameter-rich model, can we characterize when do we have dynamical instability? And so the obvious result here is, I basically already said that, so our parameter-rich model admits instability if I can choose, oops, wrong button, if I can choose R such that this S times R is Hurwitz unstable, so it has a negative value. And the interesting part here is now, well, this is rather useless in the way how it's written, but the point is essentially, so now in order to get sufficient conditions for a system to be unstable. This is actually true. The key observation here is essentially that if we take a subset of reactions and reactants out of this, we can transform, because we can choose the parameters as we want. Basically, we can choose the partial derivatives at values as we want. We can make this Jacobian matrix basically look like a block diagonal, like a permutation of a block diagonal matrix that looks like this, where we only have reactions and reactants that we choose, so some subset of this, and everything else basically vanishes. We just choose these R parameters to be small enough so that essentially whenever we have a block like this that is regular, so it has no eigenvalues with a zero real part, then we actually find a choice of the parameters where the dynamics is basically dominated by what happens in this small subset. So the idea is essentially identify subsets in our whole network, which basically means submatrices of the stoichiometric matrix that gives us the desired dynamically properties, and ideally we can do this in a somewhat principled way. The technicality that comes in there is so-called notion of child selections, which is a kind of weird term for this. What it basically means is we select a set of reactions and a set of reactants so that we get a square submatrix out of the stoichiometric matrix. So this child selection consists of a subset of reactants and a corresponding subset of reactions, such that every reactant is associated with one particular one of these reactions. So a child selection of size 3 would have 3 species and 3 reactions, where every one of these 3 species has one reaction that it's a reactant of and that we associate it with. This doesn't mean that the other 2 species cannot also be reactants of one of these reactions, but we have sort of artificially an assignment of every species as a reactant to one of these reactions. So therefore, these child selections, because we have the same number of reactions and reactants, have the same number of, so they are square submatrices, and we can also order them in such a way that every reaction, so every reaction that's assigned to one of the species has the same number. So therefore, we have in the diagonal, you always have a reactant of the corresponding reaction. And since that reactant is consumed in the stoichiometric matrix, you have, in general, a negative entry there, unless you kind of overproduce. But since we don't have a direct catalyst, we don't have to worry about that, because if something is consumed in that reaction, it's not also produced in the same reaction. So we get, in these child selections, we can, after this reordering of the columns, so after a reaction to one of the reactants in this child selection, we get negative entries in the diagonal here. So this will be the main technical construction that we need. And what's neat about this is, if you take a child selection from some submodel that's a child selection of a larger network, then that smaller child selection of the child selection is again a child selection of the original matrix. And that has the nice property to define minimal inclusion, minimal child selections, so minimal minimal square submatrices of the stoichiometric matrix with a particular property. So for instance, with the property of having an unstable eigenvalue, and that basically leads us to this notion of course property that's heritable under building child selections. There is a way of building minimal child selections, and in particular you can, for instance, define something like an unstable core, which is nice. So, and then we get this nice chemical reaction network with a stoichiometric matrix S admits instability if there is a child selection kappa. Kappa is Hurwitz unstable, so it's an unstable matrix. The way how you see this, we can choose whatever we want in this RJM, and so basically we can choose using the trick that I showed before with just making everything outside of this child selection arbitrarily small. Then we can choose, because of the design structure of this R matrix, we can just make this R matrix a perturbation of the identity matrix. So therefore, we don't really care. In ARA, choose them very close to 1 in the diagonal of the child selection and the same almost 0, so epsilon everywhere else, and we get a matrix that has a positive eigenvalue in the stoichiometric matrix exactly if we can find if this S of k, if the restriction of the stoichiometric matrix to this small quadratic plaque has a positive eigenvalue. We call this an unstable core, basically because it's the smallest ones of these are the unstable cores, and an unstable core is a who is unstable child selection for which no proper principal submatrix, which would be a child selection again. That's easy to see. Is again unstable. What one can also prove, not surprisingly, you basically have to look at small irreducible square submatrices of the stoichiometric matrix that have a positive eigenvalue. That gives you the things that can generate instability in these networks. One can go a little bit further. This is a sufficient condition, but it's not necessary. One can repeat basically just looking at the argument that I sketched very briefly before. Instead of making this matrix R just the of the identity matrix, we can also just choose an arbitrary positive entries in the diagonal and make everything else very small. So that leads us very neatly to so-called D-unstable matrices. So D-unstable matrices have been introduced a long while ago in economics. They are defined as matrices where after you multiply them, so if you multiply them with a positive diagonal matrix, you can make them unstable. What in economics is usually looked at so-called D-stable matrices, where multiplying the matrix with arbitrary a positive diagonal matrix always still has only negative eigenvalue, so always keeps them stable. So this basically just inverting this notion here, and one can immediately show that there is also D unstable core, so you can find minimum D unstable child selections in these matrices, and every unstable core has to necessarily contain either D unstable core itself, so it's the smallest D unstable submatrix, or it has a smaller submatrix that then is D unstable, and so that these D unstable cores give you the minimal matrices that make this network unstable. What we don't know is whether this condition is also necessary. So what we would like to show is that if there is no D unstable core, then you cannot have an unstable fixed point in the interior of your state space. We tortured a lot of people with who spent their lives working in linear algebra, but nobody so far could give us a hint on how to approach this or how to prove this. So I'm just throwing this out as an interesting open problem that is easy to prove, and we just too stupid to see the idea. Difficult problem, really. Yeah, here already was. Let me just skip over this feedback stuff that's I don't want to, because this is very chemically. I don't want to go into this chemical part here. The second part of my presentation is about autocatalysis. So autocatalysis is a process by which you have one compound that together with some food molecules eventually is able to produce or is converted into two waste. so it's actually an interesting thing because in chemistry there is not really one well-accepted definition of what autocatalysis actually means. Analysis agree that whenever you have something like this looks like autocatalysis, but in general you also want to have that the whole system is linked, so you cannot have just some reaction that that uses A as a catalyst and then something unrelated that produces A, and if you just look at the net effect of that, it looks like you have an A, gives you A. So this is this problem of you have a trivial sum of these reactions without kinetical coupling. You would not want to call these autocatalytic. So a few years ago, Philippe Nguyen in Paris came up with the following definition to overcome these. So the idea is basically take a submatrix of your stoichiometric matrix and multiply that with a positive flux vector. So basically you add up the through flow through all the reactions in this submatrix with some basically some arbitrary strengths, and then you will get in this submatrix you will just get a production of all the species that are used in there. So you have a net production of everything in this submatrix, and then every reaction for every reaction column there are entries in this submatrix that are used and that are produced. So you really, in this submatrix S prime, you are actually moving the material around. So the first condition says that the system can produce more of all the rows. The second condition says whatever you use up, you also produce in this system. And he defined a minimal autocatalytic core as a minimal submatrix of this type. So the question then for us, does that have anything to do with our notion of cores as minimal selections in this sense of the instability, and sort of the technical annoyance is improving this is to show that Nguyen's autocatalytic cores really are child selections, and not going into any of the technicalities there. One, do the following. So first, a little bit of notation. A matrix A is called a Metzler matrix if it has negative diagonal entries and only non-negative off-diagonal entries. I forgot to write the negative diagonal entries here, sorry about these. What we can show is that a submatrix of the stoichiometric matrix is an autocatalytic core if and only if the corresponding child selection is an unstable positive feedback, which basically unstable positive feedback thing is what I closed over. That's an additional condition on the determinant of this matrix. While the unstable negative feedbacks that are the ones that have the other sign of the determinant of SK, they are never autocatalytic. So this basically resolves two things that were interesting for chemists, because in chemistry there was a general idea that whenever you have this dynamic instability, the root cause of that chemical instability is catalysis. And basically show that this is not the case. There is actually two different ways of getting instability. One is actually autocatalysis, as everybody suspected, but there is also this other type of feedback systems, the unstable negative feedbacks, that are completely distinct from autocatalysis. And what one can even show, they can have the very same what distinguish the two types, just the spectrum on the local dynamics around the fixed points, so they are only distinguished by sort of global properties in the network. So what we get in particular is whenever we have an autocatalytic network, then it admits instability, but the converse is obviously false. And so this is just one example of such a chemical system that gives not the Metzler matrix here because it has a negative entry here, and it's the associated system nevertheless nicely produces delay, so we can have this dynamical instability without having autocatalysis and without having this Metzler matrix structure. Here's just one example with an unstable core like this, same thing. So nice complex dynamics without any autocatalysis. So in summary, parameter-rich kinetics is a nice way of getting at least some qualitative existence results on chemical reaction networks. For the chemists, instability and other interesting dynamical behavior is something that is not linked to the notion of autocatalysis, but it's actually something that's appears in much more general settings. What I think is interesting in this whole field is the notion of cores as minimal child selections that have interesting properties. They basically give you sufficient conditions for structures in your network so that you can find parameters that will generate the dynamical behavior that you're interested in. This leaves us with a bunch of interesting open questions for mathematicians interested in this type of dynamical systems. So one is what I already alluded to, whether the d-unstable cores are necessary for the network. So if we can prove that we've essentially already achieved a characterization in terms of the existence of instability. It seems this is something that Nicola and I are working on in the moment. Can we also do similar things for limit cycles and related stuff? So Nicola already had some very interesting results in that direction that is in part already published. The final question also maybe more for chemistry-minded people, or an actually surprisingly large community who is interested in the dynamics of mass action kinetic systems from a very mathematical point of view. How does mass action kinetics fit into this? Can one characterize the unstable cores do not meet instability under mass action kinetics? So if you go to the limit of mass action kinetics, the possibility, the freedom to make the subsystem unstable disappears because the system essentially. Is this something that one can characterize? So one could hypothesize in this a point, for instance, that it's only a fairly small subsystem where you have this problem. If they are large enough, you still have enough parameters essentially to drive them to instability anyway, even if you don't have the full degrees of freedom that we have in these weakly monotonic systems that we're using here. So, but it may be there is still, even if the system is not parameter rich, there is sufficient parameters to still enforce instability if the systems become large. And for small one, you have so many constraints that it doesn't work. But I have no idea if this works. And I also don't know how to approach this except for, going through lots of case by case steps. Yeah, with that, I'm at the end of my presentation. Thanks for listening. What I have to do at this point. Of course, I have to acknowledge, in particular, Nicola Vasena, who is a researcher in our lab in Leipzig, who came with his own money as a kind of senior postdoc to work on this topic with me.
Okay, thank you very much, Peter, for your talk. There are some questions.
Hi, a nice talk. But this is just a curiosity. Is there any relation of this with this notion of controllability that appears in linear control systems? Because in some sense, this idea of changing the stability, changing the eigenvalues of the matrix using parameter also happens in that theory. Do you know about that or not?
I haven't thought about that at all, but that's certainly an interesting angle that is worth thinking about.
Yes. A second question. You talked about negative and positive feedback in this for these systems, right? But more or less, what's the definition of negative and positive feedback?
So the definition of that essentially, let me just go to these slides. Where are they? Sorry, I'm very bad at remembering them. So an unstable positive feedback is an unstable core that in addition satisfies that the determinant is negative for even-sized number of species, and it's for an odd-sized. It's positive for an odd-sized number of species in the subsystem, and you have the for the unstable cores you have the you have the opposite sign of the determinant. So you basically can think of that as one way of generating these, just in terms of a feedback network, is you have in one case you have only positive feedbacks, in the other case you have only negative feedbacks. There would be examples of these.
Thank you.
More questions? I have a question. Sorry. Okay. You mentioned in the first slides that in your networks, the nodes of your network, the molecules have some label. This label is always known, or you have some uncertainty about this?
So
the labels.
I mean, normally we know what these labels are, because they are specific chemicals, and the assumption essentially is, so for say an enzyme reaction or so, we know what goes in as reactants and what comes out. This is, of course, depending on your application, not always true in reality, right? So you will have enzymes that are promiscuous, as they say, in terms of the reactants that they can process. So there's many enzymes that actually can have more than one substrate that goes in there, and then you basically, but that you would essentially model as I have a number of parallel reactions, and it's just a reaction mechanism that's exerted by the enzyme that is constant, that's given by the enzyme, and but so at least in this dynamically systems context, you essentially always assume this underlying hypergraph is given in a deterministic way.
Okay. Because we have some project about community detection in networks, and I have a PhD student working on exactly in hypergraphs, community detection on. So I wonder if this could be an example where maybe you want to
Yeah, so maybe for community detection, if you basically are interested in how to carve up, so one other way of looking at these systems would basically be you have all these little subsystems in there, and can you cluster that? Is there a way of coarse graining
Yeah.
systems like these? So in terms of you have large reaction rates inside, small reaction rates outside,
More or less.
and then so in this coarse graining sense, I can immediately see that ideas of community detection also could be useful. On how much you can say about the dynamics in such coarse graining settings is another issue. So normally this only works if you really have different orders of magnitude of rates inside and outside, so that you can say something, and you need irreducibility inside. Otherwise, you only get transient behavior, and you can't get anything in a steady state setting, for instance. So, but it's definitely an interesting point or an interesting angle to look at this.
Okay, thank you.
Thank you. I don't know so much about chemical reactions, but I was imagining something like a network flow. For example, you consume something and you produce another thing, and everything is positive, and you have this kind of reaction or transfer of information. And in this sense, do you think that we can apply it, for example, in brain networks, that you have a kind of information flow, and you can use your approach, for example, to estimate the parameters of an.
So, I mean, this stuff back here, this R of X, you really can think of that as fluxes or of flows. Yeah. So they actually do satisfy conservation conditions at every intermediate. At least if you put a steady state solution here, right? So there is I think whenever you have a sufficient number of conservation laws that you can argue that what you have is really a flux that behaves like a flux. So you have nodes where influxes match out fluxes. I think techniques like that would actually work.
Oh, okay. Thank you very much.
Okay, more questions? Okay.
Do you have any way to test, like, for conditions to have limit cycles there?
We haven't really worked these out, but in detail. But what you can do for limit cycles is you can specifically look at whether the child selections allow for Hopf bifurcation. So this is work that Nicola did without me, so I know he has some cool results in that direction. Some of that's published already.
In principle, you could define the conditions that is needed for the child to have.
I would think so, but we haven't worked it out.
More questions? No? Okay, so let's thank again, Peter.
Thank you.
Okay, let me introduce our next speaker. It's André Fujita from the computer department here at IME, and he's talking about a statistical framework for random graph models. So thank you very much.
Thank you very much. Okay, so I want to present something about a framework to work statistically work with random graph models. Okay, usually when we imagine a network or a graph, we the current view, how we analyze graphs nowadays is like something like that. You have a network, for example, a functional brain network or a protein-protein interaction network or something like this. And we analyze some characteristics of this network. For example, robustness, modularity, centrality, motifs, etc. But there is a problem when we analyze in this way. For example, when you are analyzing a network, for example, suppose that I want to compare my functional brain network with Aline's functional brain network. And then in this case, suppose that we have, for example, same number of edges, or a little bit different number of edges, or same centrality, or same robustness, or any other characteristic. So in this case, can we say that our functional brain networks are the same or not? I think this is the point, because everyone here, we are all different. So we have a kind of, how can I say, not only, it's not perturbation, but a kind of noise, that this noise can come from the measurement, for example, your technique that you are measuring your functional brain network, like fMRI, or the construction of your network, or also because it's unique. Each one here is unique. So I think it's very, very difficult to compare functional brain networks or any other kind of biological network. For example, if I compare my metabolic network or all my chemical reactions in my body, in my cells with Peter's chemical reactions, probably it will not exactly be the same. It will be a little bit different. But if we suppose that we belong in the same group, it's nonsense because, well, if we are in the same group, probably we have a kind of the same network. It's just a kind of noise that you have in the network. So the first thing I think is it's very important to define what the networks are the same. And in my point of view, I think, thinking in a statistical manner, I think two networks are the same if the generation mechanisms are the same. For example, in statistics or in physics, when you say that two objects are the same because usually they were generated by the same process. For example, you have two sets of numbers, two groups of people, and you have their weights or their heights. So, of course, they are not exactly the same. But if you think in a statistical manner, probably if they were generated by the same model and same set of parameters, you can say that they are the same, okay? So what we propose here is not analyzing the network itself, but the generation mechanism that generated this network. So instead of analyzing my functional brain network, what I propose here is to analyze the mechanism that generated my functional brain network. So again, like in statistics, we can infer some properties, for example, about develop some methods for model selection, parameter estimation, classification, correlation, causality, etc., 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 like in a statistical fashion. But here we have the second problem. The generation mechanism is rarely unknown. For example, do you know what the set of mathematical equations that generated your functional brain network? Do you know a random graph model that generated your functional brain network? I think, well, at least in my point of view, we don't have, at least now, a good model that fits very, very well to model functional brain networks, and neither for metabolic networks or protein-protein interaction networks and social networks. So how can we do inference? How can we infer from generation mechanism if we don't know the next generation mechanism? In other words, it's not observed generation mechanism. So the proposal here is, oops. So the proposal here is to analyze the graph spectral distribution that I think it's a good proxy for the generation mechanism. So what's the graph spectral distribution? Suppose that you have a graph, and you have the adjacency matrix. So suppose here, if you have nodes 1 and 2, they are not connected. So row 1, column 2, you put 0. And if they are connected, for example, node 1 and node 3, you put number 1 in column row 1 and column 3, and so on and so forth. You obtain the adjacency matrix. And in a very caricatural fashion here, suppose that this graph is very, very large, and so you have a very large adjacency matrix. And you can obtain n eigenvalues. You can decompose it by eigenvectors and eigenvalues, and you select the eigenvalues, and you construct the distribution of the eigenvalue, probability distribution of the eigenvalues, and you obtain the spectral distribution. And why do we analyze the spectral distribution? Well, here's the rationale. For example, you know that if you have a graph composed of n nodes, you have n eigenvalues. So the dimensionality of what you are analyzing is much fewer, because if you have a graph, you have n nodes and at most n square edges. In this case, you have only n eigenvalues. So it's a kind of dimensionality reduction too. You can think about a kind of dimensional reduction. The graph spectral distribution is also related to the structure of the graph. So you can find, for example, partitions of the graph, or the largest eigenvalue is a kind of function of the parameters of the random graph model, etc. So you can infer several kinds of structures of the graph. And so on. And what we did almost 10 years ago is to describe what is the graph spectral entropy. So it's a very naive approach that we just applied a kind of the channel entropy for the spectral distribution, and we'll obtain this definition of graph spectral entropy. And what's the interpretation of this graph spectral entropy? Essentially, suppose we have an edge-linear random graph. Edge-linear random graph is essentially you have a pair, for each pair of nodes with probability p, you connect them with an edge, okay? So here in the x-axis, if you have probability p equals to 0, you obtain an empty graph. And here on the right side, if you have p equals to 1, you obtain a complete graph, okay? So in this both cases, you obtain the lowest entropy. And in the center here, at p equals to 0.5, you obtain the highest entropy. And I think it's a little bit intuitive, because, well, which kind of graph you have, which graph is the most random? It's probably when p equals to 0.5, okay? So this is the equation that we derived for the graph spectral entropy, and this is the analytical function for the graph spectral entropy. So I think it's a little bit intuitive that it's the most unpredictable graph. Well, if it's unpredictable, it has more graph spectral entropy. If it's more predictable, the graph spectral entropy is lower. But unfortunately, at the moment, we don't have we couldn't derive a relationship between the graph spectral entropy and the structure of the graph. This is a second example, the Watts-Strogatz random graph model. You start with p equals to 0, you have this kind of circular graph, kind of regular circular graph, and as you increase p, the probability p, you shuffle each edge with probability p, and with p equals to 1, you obtain a complete random graph, like our edge-linear random graph. And you can see here is that as you increase p, you obtain higher values of graph spectral entropy. So as you increase the randomness of the graph spectral entropy also increases. And there are other types of random graph models. It's just for illustration that our intuition is in accordance with the randomness of the graph. And these two figures is just to show an illustration of the application of this method, that we could use the definition of graph spectral entropy to identify new brain areas associated with ADHD in the left side and autism spectrum disorder in the right side. That in the left side, we obtained this green area, mostly composed by the somatomotor region. And here in the right side, in autism, we identified the cerebellum, that is this orange side that has the graph spectral entropy increased in cerebellum in autistic children. Okay, and now we will start with some methods to do inference from the random graph models. In basic statistics, we know, for example, the maximum likelihood estimator. Suppose you have a data set, you have a model, a statistical model. In this case, you have the normal distribution. In this model, you have two parameters, the mean mu and the sigma squared, the variance. And we can estimate these two parameters using a maximum likelihood estimator, right? And for graphs. You observed this graph. I tell you that this graph was generated by a random graph model. So it's equivalent here that you have this data set, that is the graph. You have the random graph model, okay? And you want to estimate the parameter P of the edge-linear random graph model. So in this case, it's very easy because the maximum likelihood estimator is just to count how many edges you have here in the graph and divide by the total number of possible edges. So you obtain the parameter P, estimate the parameter P, that's 0.51. That's very close to the parameter that I used to generate this graph, that was 0.50. Okay, in this case, it was very, very easy. But and for what is Strogat's random graph model? You have this regular graph, and with probability P, you shuffle each edge of the graph. So how can we estimate this P? In this case, I think it's much more difficult. I don't know in the literature a good parameter estimator for this graph. Sorry. So how can we estimate this parameter P? So the trick here is to use the trick used by Ronald Fisher. We don't have the likelihood function for random graph models. Usually we don't have, okay? So in this case, we replace the likelihood function by the spectral distribution of graph. I mean, we use it as a proxy for likelihood function. So in the left side, you have the observed graph, and the right side, you have the model. You can generate the spectral distribution of the model. If you don't know analytically the spectral distribution of the graph, you can generate it by Monte Carlo simulations. So you can use the algorithm to generate the graph many times, for example, 30 times, and take the average and use this one as the spectral distribution of the graph. And then we estimate the parameter theta here that minimizes the distance between the random graph model and observed graph. So it's exactly the same idea of Ronald Fisher and maximum likelihood estimator. So the parameter theta that minimizes this distance is exactly the estimated parameter, okay? Well, here we use the Koba-Kleiber divergence, but in practice you can use any other kind of distance. For example, later I will tell you that we replaced it by the L1 norm and it worked better. So this is just an illustration here. To show that we have, each row we have different random graph models. Between parentheses you have the parameter, and each column you have the size of the graph. And as you can see, as the size of the graph increases, I mean the number of nodes, you will obtain better estimates for the parameter, okay, using this approach. So for some of them, like Watson-Strogat's random graph model or the Barabas-Albert model, that we don't know analytical parameter estimator, using our approach, you can obtain very good estimates for them, okay? But as you note here, it's very computationally expensive. Why? Because, well, if the graph is small, it's fine. But if the graph increases, for example, you have n nodes like 1,000, I think you can do it in a laptop. But if it becomes 1 million nodes, it becomes unfeasible in laptops because you need to calculate all eigenvalues values here and construct spectral distribution, and you need to do it many, many times for each parameter theta here. So it's very expensive, I mean, computationally expensive. So what we did here is in collaboration with Professor Peter Stadler here, we developed some algorithms also with Grover, that is my postdoc here, many algorithms to efficiently approximate spectral distributions of graph. Of course, it's not a general method, but for some particular characteristics of the graph, for example, if it's locally tree-like, or if you have short cycles, we can estimate them very efficiently, I mean much less than any cubic. For example, if you have a locally tree-like, we can do it in a linear time and space. We also replaced the Koba-Kleiber divergence by the L1 norm, and this part we could prove that our estimate to converge exactly where we want to converge. And then we obtain some interesting results, I mean computational interesting results. For example, if we simulate a Barabas-Albert model with 5 million nodes and average degree of 5 and parameter 1.15, if you use our previous approach, it's infeasible. You probably will need 900 a year, so it's impossible. But if you use our optimized approach, you can do it in 2 days. Well, you can say, okay, 2 days, 48 hours on average is too much. But you think that, you cannot do it, I mean, analytically, neither using a conventional approach. I think 2 days is something that you can wait. And we are not saying about using a supercomputer here or GPU. It's in a simple workstation, okay? An advantage here of the approach developed by Grover is that it only needs linear space. So if you think that the graph, you need a adjacency matrix, so it's n square, you need only linear space. So you can allocate memory for 5 million nodes, okay? But it's impossible if you need the square matrix. Okay, second approach is model selection. It's very similar to the parameter estimator. Suppose that you have a data set, you have 3 candidate models here, and we want to choose which candidate model is the best one, okay? Following a certain criterion. Here, I think it's very simple that you, by looking at these 3 models, you can say that here in the center, you have the most appropriate model because it's not overfitted like in the left one, and it's not too simple like the right one, okay? So one of the criteria, there are many criteria here. For example, the archaic information criteria is one of them that we based our approach on this criterion. Here again, you have the likelihood function here, but for graphs, we don't have a likelihood function. So what we did is, suppose that you have three or more candidate models. You have the spectral distributions for each one that you can again simulate by Monte Carlo simulations. And we choose the model, and you have to observe the graph. For example, your functional brain network. So suppose that I have my functional brain network here, observe the functional brain network, and here we have 3 candidate models. For example, the first one is was constructed by Aline, Peter, and I, for example, and which one is the best one? So I select the one that minimizes the Koba-Kleiber divergence and penalize it by twice the number of parameters. Of course, because if you have a more complex model, you have more parameters, and it's easier to fit to observe the graph, okay? So you need to penalize by the number of parameters. So we have very similar to the archaic information criterion. We have a kind of archaic information criterion for random graph models. But we just replaced the likelihood function by the spectral distribution. And this is just an illustration. You have seven or 8 protein-protein interaction networks here. And we select our candidate models are 3, Erdős-Rényi, scale-free, and small world, I mean the Watt-Strogatz. And our approach select the scale-free model as the best one. It does not mean that the scale-free network was the model that God used to generate these graphs, but it only means that the scale-free network is the one that best fits your data. But of course, in the next year, for example, if another guy appears here with a 4th model, maybe this 4th model fits better than the scale-free, okay? Another approach that's very common in basic statistics. You have, for example, 2 data sets, for example, height of guys and girls, or weight between guys and girls, etc. So you have 2 groups, and you want to test if they are equal or not. So you can test, for example, the median, or the mean, or the maximum, etc. In this case, usually people test the average of group 1 versus the average of group 2. And how you do it? It was developed already by William Gossett, the t-test, that you have 2 distributions here, and you want to test if the means of these 2 distributions are equal, also by considering their variability, I mean the variance of the data set, okay? Now suppose that you have 2 data sets of graphs, for example, functional brain networks of control people, and another group of people, functional brain networks of children diagnosed by, I don't know, ADHD or autism or something else. And we want to test if these 2 data sets, 2 groups of networks are equal or not. In other words, if these 2 groups of networks were generated by the same model and same set of parameters, okay? Because of course they are a little bit different. If you count the number of edges or centrality measures, of course they will be a little bit different. But we are interested in if the generation model and the set of parameters are the same or not, okay? So again, we use the spectral distribution. So you suppose that you have many graphs here and then another set of graphs here. You calculate the spectral distribution of each graph, calculate the average, I mean the mean spectral distribution for each one. And we want to test if the mean spectral distribution of this set of graphs and the mean spectral distribution of the second set of graphs are the same or not. To do it, we calculate just the divergence between them, like the Jensen-Shannon divergence, that essentially is a function of a Kuba-Kleib divergence. But the advantage that it is symmetric, and also the triangular inequality is valid in this case, okay? And the hypothesis test. So if the distance between them is equal to 0, so you accept the null hypothesis. Otherwise, you have statistical evidence to reject the null hypothesis, okay? So this is just an illustration to tell you that we compared here. I don't remember exactly the numbers, but I think it was more or less about 500 children diagnosed with ADHD and 500 children, control children. And we showed here the p-values. And if we test the spectral distribution, as I showed in the previous slide, we obtain a more significant statistic than the other approach. For example, if you count the number of edges or centrality measures, etc. Okay, well, this one is just an application in bioinformatics, I mean gene expression data. I will skip this slide. And if we could construct a t-test, I mean, if we succeeded in constructing a test to compare two data sets, can we generalize it for K data sets, like in the analysis of variance? And the answer is yes. What's the analysis of variance developed by Ronald Fisher? You have 3 data sets or more, I mean 2 or more data sets, and you want to test simultaneously all of them. So given two or more data sets, all means are equal or not? This is the question, okay? And for graphs, exactly the same. You have 2 or more data sets of graphs, and you want to test if all of them were generated by the same random graph model, or at least one of them were generated by different random graph model, okay? So the generalization is very straightforward. So you calculate the spectral distribution for each graph, and then take the average for each population of graph, and calculate the average of averages, and calculate then the Kuba-Kleib divergence between each population to the average of average. So the intuition is, if at least one of them is different, so probably one of these Kuba-Kleib divergence will be greater than 0, okay? Otherwise, if all of them were generated by the same random graph model and same set of parameters, all of them will be equal, and of course, the average of averages will be also equal. So probably this Kuba-Kleib divergence will be all 0. I mean, almost 0, okay? So this is the intuition of this approach. And what we applied it for fMRI data of autistic children. So we had 3 groups, controls, Asperger, and autistic people, okay? We tested each subnetwork here, represented by different colors, and what we obtained was significant statistics for the cerebellum again. So the question is, because first we tested the controls versus Asperger versus autism, okay? And then we obtain a significant p-value here. But the next question is, which pair is different? I mean, controls versus autism, controls versus Asperger, or Asperger versus autism. And then we did the pairwise test, and we obtained that the controls as autistic children are significant here. And we didn't observe any kind of statistical evidence for controls versus Asperger and Asperger versus autism. That I think is reasonable, because nowadays Asperger belongs to the group of autism. But 1 decade ago, there was 3 groups. And Asperger was a kind of in the middle between controls and autistic children, okay? And you can say, okay, Fujita, you said that you have many graphs here, but usually we don't have many graphs. What you have is only one graph. For example, you have one social network, one Facebook network, okay? Or you have one LinkedIn network. So you don't have replicates. I think in functional brain network is a kind of a particular characteristic of this data set. We usually have only one network. So how can we test if, for example, if the social network, I mean, for example, the Instagram network or Twitter network of Germany was generated by the same set of parameters and model of the Brazilian LinkedIn network. So At the first glance, it seems to be impossible because you don't have replicates, and usually in statistics you need replicates, okay? But now suppose, just to give this intuition, suppose that you have the Edge-Renyi random graph, okay? Edge-Renyi random graph model. You can estimate the parameter of the Edge-Renyi random graph model by just counting and divide by the number of total edges, right? And you know that it was each edge was follows a Bernoulli, and then you have a set of Bernoullis, so you have a binomial distribution, and you can estimate the variance of the parameter, right? I mean the probability P, because it's a binomial distribution, okay? So if you have the estimated parameter and the variance of this estimated parameter, you can test it if two parameters are equal or not, okay? Like using a t-test or analysis of variance. So you can test two random graphs, okay? For example, the Edge-Renyi random graph is a very simple example, but if you can estimate the parameter of the graph, and also you can estimate the variance of this estimated parameter, you can test them. So I think it's possible to do it. So the trick here is to use our parameter estimator that I showed you in the previous slides, and then via bootstrap, estimate the variance of the estimated parameter, and then the rest of the algorithm follows the analysis of variance. Okay, it's exactly the same as the analysis of variance that Ronald Fisher already developed 1 century ago. Okay, so the only one point here is that, well, we use the F statistic here, so we couldn't prove it mathematically that the estimated parameter follows a Gaussian distribution to use this F statistic. But by simulations, we show that this distribution is very similar to a Gaussian distribution, a normal distribution. And by simulations, we also show that the control of the false positives rate here using this statistic is very good. I mean, it follows, for example, if you have a p-value threshold of 1%, the number of null hypothesis rejected is approximately 1%, and so on and so forth, okay? So it seems that it gives us a very good approximation. And it's just application in protein-protein interaction networks. Okay, another concept is about correlation. Suppose you have scores in physics and score in maths, for example, X and Y. So you have, for example, 100 students, and you plot, you construct here a scatter plot, and you can see here that if you know the score in maths, you have a very good hint, a very good approximation of the score in physics, okay? This is what intuitively we know as when two data sets are correlated, are linearly correlated, okay? So this is the idea of the correlation. But now the question is, and about graphs. You have two vectors of graphs, X and Y. Suppose that the X is my functional brain network, and Y is Aline's functional brain network. Can we tell here that these two vectors of brain networks are correlated or not? But by just observing these two vectors of networks, I think it's very, very difficult to say they are correlated or not. So the first question is, what's correlation between graphs? Okay? And the idea here is, suppose that each graph was generated by a random graph model and its respective parameter, okay? If the parameters of the random graph model are correlated, I say that the two vectors of graphs are correlated, okay? So in this case, these are in red are the parameters of the graphs, the random graph models. And here, if you calculate the correlation between the parameter of graph X and the parameter of graph Y, you observe here very linear relationships. So I can say that these two vectors of graphs are correlated, okay? So by just observing the graph, I think it's very difficult to say that they are correlated. But by analyzing the parameter of the graphs, the random graph models, I can tell you that they are correlated. Okay, but we have a problem here again. We don't know the parameter of the random graph model. So, because if you know the model, you can estimate it using our approach, right? But if you don't know the model, what you can do next is you use our selection model approach, but you need, again, a set of random graph models that you think that one of them may fit well our graphs, okay? But in the worst case, you don't have the random graph model, neither parameters of the graph. So How can we solve this problem? The trick here is to use the spectral radius, I mean the largest eigenvalue of the graph. Why? Because the largest eigenvalue of the graph, for most random graph models, I don't know if it's for all of them, but at least for the majority of random graph models that at least I do know, the largest eigenvalue is a function of the parameter of the graph. For example, for the edge-linear random graph model, it's essentially the probability P times the number of nodes. Okay. For the K-regular graph, you know that the largest eigenvalue is exactly K, I mean the degree of the graph. And for other random graph models, we don't have analytical formula, but by simulations, we can see here that there is a monotonic relationship between the parameter and the spectral radius. So it's essentially a function of the parameter of the graph. So we can use the spectral radius as a proxy for the parameter of the graph, okay? So essentially, in summary, the correlation between vector of graphs is essentially the correlation between the largest eigenvalues of the graphs. Okay. Of course, we proved that our approach converges, and we have something like a Spearman correlation between random graph models. This is just an illustration here. We compared the correlation between for 5 parts of 5 subnetworks of the function brain networks of control children against ASD autism children, and these 3 figures shows when this correlation changed between controls versus autistic children. Okay, so in this between default mode and visual control versus default mode and default mode versus somatomotor, we can see here. Sorry, I didn't tell you. The red line here is the autistic children, the blue line is the control children. And we can see here in these 3 cases that the correlation between these functional brain networks increased in autistic children than controls. Okay. And the same idea is for Granger causality. Instead of correlation, now suppose that you have 2 time series of data. And what's Granger causality? Very briefly, if you can have a good estimation about the future of this dashed time series using the past values of the solid line, using passive values of the solid line, you can say that the solid line, I mean the X time series, has a good predicts well the future values of time series Y. So in other words, X Granger causes Y. The intuitive idea again is like the cause always happens before its effect, right? At least in our universe here. I don't know about other metaverse, etc. But in our universe here, in this Newtonian world, we know that the cause comes before its effect, right? So I think it's very intuitive. So essentially is, if I analyze passive values of X, can I predict future values of Y? If yes, there is a kind of causality between from X to Y. It's not causality, I mean a kind of Aristotelic sense, in philosophical sense. Why? Because in weather forecast, there's a very useful example. For example, in weather forecast, we say that tomorrow will snow, and it snows. Tomorrow will rain, it rains. So the guy of the weather forecast is causing weather of tomorrow? Of course not, okay? So it's a kind of hint of causality, but it's not causality in an effective way, okay? Again, the trick here is you have 2 time series. Now you have the time here. You have 2 time series of graphs. It's changing over time, the structure of the graph. If you analyze the structure of the graph, I think it's very difficult to say if one of them is causing the other. But again, you can analyze the parameters of the graph. I mean, the largest eigenvalue again. And, well, if you analyze the correlation here, you cannot see any kind of correlation between them. But if you shift a little bit one of the time series, you can see a linear correlation. So, I mean, for example, the t minus 1 of X is correlated with YT, and so on and so forth, okay? So in this particular case, we can say that the time series of graphs X is Granger causing time series of graphs Y, okay? This is another example. It's an illustration again in autistic children. So we divided the brain between left and right sides. So you have left and right functional brain networks. You have 2 time series of brain networks. And we tested if the left side is causing the right side or right side is causing the left side differently between controls and autistic children. And what we observed that yes, in fact, yes, these 2 regions of the right functional brain network is causing the left side, okay, particularly in these 2 areas. This result was not published, it was just a toy model. We have 2 violinists here and we collect the data, it's F-years data, while they were playing violin. And we observed that the violinist A was Granger causing violinist B. In particular, in this case, well, you have the data of both of them. Violinist A is 50 years old, 40 years of career, right-handed. Well, both are right-handed. And violinist B is 41 years old and 28 years of career. And we can, and they, apparently they were playing together, okay? So it was very difficult to note any kind of delay in terms of sound, okay? But when asking them, we know that violinist A is more senior than B. So, well, I'm not a musician, I don't know play any kind of instrument, but it seems that, well, they, how can I say, they did before something like the junior guy follows the senior guy. The question is there any kind of eye contact or movement or sound that there is this kind of Granger causality? Because it's not like the force, okay? So probably there is a kind of mechanistic explanation for this. What we did was analyze, first of all, the movement, because probably there was no eye contact here. So probably this kind of information flow was based on the movement of their hands. Probably violinist B was checking the hand of violinist A. So we did a kind of movement track here. But unfortunately, we couldn't test it because we lost their hands during the movie. So we couldn't test it. But probably we suppose that it was the information flow probably was their hand movement. Okay, unsupervised classification. Usually when we talk about graph clustering, we talk about something like this. Suppose that you have a graph and more connected nodes belongs to a cluster and sparsely connected nodes belongs to different clusters, right? But the problem that we want to solve here is different. Suppose that you have a set of graphs and I want to cluster similar graphs in the same cluster and different graphs in different clusters, okay? So it's a little bit different, the problem. So again, in basic statistics, what we have here is the classification expectation maximization algorithm. Let's suppose that you have each cluster is represented by a model, for example, a Gaussian distribution. So you have a data set. What we want to do is to check each item from which probability distribution it came. For example, you have 100 numbers here, and you say that there are 3 clusters, for example, 3 Gaussian distributions. And what I essentially do is for each item from what's the probability that it came from Gaussian distribution 1 or 2 or 3, okay? So here the definition of cluster is very clear. It's the number of Gaussian distributions and each Gaussian distribution represents 1 cluster, okay? We do the same for the graphs. Now suppose that you have a set of graphs and you have a set of random graph models. So you know a priori, well, you know what's the number of clusters and you need just to estimate from which random graph model each graph came, okay? So the algorithm is essentially the expectation maximization algorithm for clustering. This is the illustration. We have Chibi1, Chibi2 are, it's essentially the same monkey, okay? We repeated the experience twice. Well, we know the Japanese people. And well, you put the monkey in a chair and you collect their electrocorticogram data for 30 minutes. I mean, here, in this green line, they inject a drug and the monkey sleeps, falls asleep on, well, where is it? Observer anesthetized, sorry. Yes, here the anesthetic injection is to observe when the monkeys didn't respond to the medical doctor, I mean to the researcher. And here is when our approach, our clustering approach said, okay, from here now the monkey is not responding. I mean, his brain is not responding, okay? So what we did is we collected the functional brain networks and we said, okay, there are at least two states. I mean, the monkey asleep and sleeping, awake and asleep, and we clustered them. And we take the average here, and so we injected the drug here. It's when the medical doctor said the monkey is already anesthetized, and here's when we predicted it, okay? So it's after the injection, before when the medical doctor confirmed it. You can say, of course, it's obvious, this result. But if you use any other kind of clustering algorithm or classification, usually sometimes this dashed line comes here, that's impossible. And sometimes it comes here, that's useless, okay? And same for when the monkey is awakening here, that here is observer recovery, and here is when our clustering algorithm said, okay, the monkey is already recovered. So it's just to illustrate that sometimes it's better, the phenotype that monkey is sleeping or not is later than when if you analyze the functional brain network directly, okay? And supervised classification. The last one, suppose that you have a data set and you want to classify if they are squares or circles. Okay, so suppose that you have functional brain networks, people diagnosed with disease and control people, and you want to train your AI, I mean, in this case, is the linear discriminant analysis, and obtain a classifier. But for graphs, what we did, in this case, Grover again, the postdoc is sitting here, he extended linear discriminant analysis for graphs. Essentially here, what he did was to use the spectral distribution again as a proxy for the graph and construct a kind of functional linear discriminant analysis, okay? So our motivation was to construct this classifier for graphs, but then when we submitted the paper for the journal, one of the reviewers said, okay, well, you don't have a classifier for graphs, but you have a classifier for general functions. So we changed a little bit the introduction of the paper. And what we have here is many time series data sets, and we obtained that our approach, our proposal obtained the lowest error. And it's a second application that we show that the Grover's approach is very interesting because instead of obtaining just the classifier for the time series data, he can also tell us which frequency of the time series function is the most important one for classification. Okay, all of these approaches are already implemented in R or partially in C and are in the StatGraph package that's freely available. And just one thing more that I would like to tell you is, usually people ask me, what's the difference between our approach that is based on this network statistics and artificial intelligence is the boom nowadays, and for example, graph neural networks. And what I want to tell you is that when your approach is based on artificial intelligence on graph neural networks is that it's excellent. It's very, very good for predictability, I mean prediction. For example, I usually compare it what Tycho Brahe and Johannes Kepler did centuries ago. because Tycho Brahe, he collected the astronomical data set, a very big data set at their time, and then gave it Johannes Kepler just before he was dying in his bed. And then Johannes Kepler found, without AI, I mean, only used the original intelligence, the patterns for the 3 laws of Kepler. But of course, these 3 laws of Kepler provides a very good predictability, but does not explain why it happens. And 1 century later, well, Isaac Newton, he appeared with his scientific revolution that with a few number of equations and parameters, he can model your natural event very well and explain how the variables related to each other. So you can explain why you observed this event. Well, at least in my point of view, I think this was the scientific revolution. I don't know, probably each one has your own point of view. But I think, at least in my point of view, this was a scientific revolution that you could explain the causality or the mechanisms behind the natural event. So what's our focus here is not prediction of the event, but we want to do inference from the generation mechanism. So we are interested in explaining the mechanisms of the event. So probably we are not good as well as graph neural networks because our prediction is not so good, but we at least try to explain the mechanism, okay? And on the other hand, graph neural networks they are very good in prediction, but they cannot explain the mechanism. So I think they are complementary. So we are not competing to each other, but we are working together. Okay. Okay. So thank you very much. These are my 2 emails. So if you have any question, you can contact me by email. Thank you very much for your attention.
Okay, thank you very much, André. There are some questions. Yes.
First of all, thanks for the great presentation. And I would like to ask you something. Some of the data that you showed just shows that you have better results when you have more nodes in your network, okay? But me, for example, particularly work with EEG data in which I have few electrodes. So I was thinking, how can I apply this? And maybe we can have a node that captures the activity of something, but in different points of time in order to have more nodes. Do you know if we can apply this framework using this kind of workaround, or there will be some constraints that will not make it possible?
You mean when you construct your functional brain networks based on EEG data, right?
Yes.
Yes, it depends on how many channels you are using your EEG data. For example, if it's only 24 or...
Yes, I'm working with 32 electrodes,
22,
so it's
okay.
very small.
Yes, because, well, 22 nodes, I don't know if it's enough, for example, because, well, we need to estimate the spectral distribution, right?
Yes.
So if you have 22 channels, you have only 22 nodes and 22 eigenvalues.
Yes.
So, well, it depends on your question and how, because I know that the EEG data is very noisy.
Yes.
So it depends also how noisy it is. And how you construct your functional brain network. Use, for example, partial directed coherence or directed coherence.
Yes. Basically, I am looking right now in some possibilities for building it, so I can have the temporal series and computer correlation, but I can use Granger causality, I don't know. But the problem here is that I have few nodes. And maybe we can do a workaround here. Ítalo will talk a little bit about his presentation, and he made a workaround that is interesting in order to be able to have more nodes within fewer electrodes, for example.
Yes, I think also if you can more precisely construct your network can help. For example, instead of correlation, you could use Granger causality in the frequency domain and filter out many frequencies that you are not interested in.
Yeah.
For example, if you are interested in, I don't know, in the alpha band or beta band or delta band, etc., you can focus only on these frequencies and filter out all the other bands, for example, and then you can obtain more interpretable and more accurate networks.
Okay, it's an interesting idea also. Thank you.
Okay, thank you.
In your analysis, you use the adjacency matrices. Why not the Laplacian matrices?
Yes. Yes, I think there are many advantages of Laplacian matrix because you know that the support is between 0 and 2. And in our case, we don't know the support, a priori at least. So yes, I think we can replace with the Laplacian matrix. I don't know because, well, everything that we proved here are for the adjacency matrix. Of course, you need to reprove for Laplacian. Okay, but my intuition is that, well, it will work as well. Yes.
Okay. Another.
Maybe it's a naive question. I don't have any kind of intuition about how because what you are looking at is the distribution of the aging
eigenvalues distribution. Yes.
of your metrics of adjacency, right? So in order to have a very good approximation of your distribution, you need very big graphs in some sense, right? You have big N or not?
Well, it depends on your definition of big, but I think 100 is enough usually.
This would be my question, because usually we were working with EEG data, so at most we will have 128 nodes. So you think even with only 100 nodes we would be able to see this distribution, and how this would characterize my graph? I don't have any intuition about how this is implying in the distribution of the graph, not the distribution of the...
Yes, I think it depends. As I was talking to him, because the EEG data, the main bottleneck, I think, is because it's very, very noisy. So if, for example, if it's, suppose that you have a group of people, for example, 10 people, and you expect that the EEG data, or at least the functional brain networks derived from the EEG data, should be similar among them, and they are not. So in this case, probably the EEG data is so noisy that you cannot see these similarities among them. So this may be a problem, but we need to check it. But I think 128, I think, is enough to have a good estimate for the spectral distribution.
And just another very naive question: do you have any intuition about what should happen with a distribution of some graph in the brain? I mean, how would it look like these distributions? It's just a guess, I think, because...
Yes. It's very difficult to answer your question, because what we are noting nowadays that depends on... I think it depends on the brain region and also the brain species. For example, we calculated the spectral distribution, I think it was very recently, 1 month ago, of the C. elegans. And it was completely different of the human. But yes. So probably it depends on the species and which region of the brain you are analyzing to. And how you construct your brain network. If it's, in fact, between neurons, or it was estimated based on EEG or functional brain network or DTI or any other kind of technology. I think it changes.
But usually it's not similar with any kind of random graph, classical random graph. Oh, okay.
No, I don't think so.
Okay, I I have a lot of questions, but I think because thank you for your talk. You talk about a lot of things, and so I have a lot of questions. But maybe it's better to go for a coffee, to do a break of a 15 minutes break, and we can talk on the coffee. Okay? So who is online? We come back in 15 minutes. Okay. Thank you, André. Thank you. Let's begin the second session. So our next speaker is Ítalo Pinto from University of California at Irvine, and he's going to talk about symbolic dynamics of joint brain states during dyadic coordination. So thank you very much to be here, Ítalo, and good talk.
Thank you very much for the invitation. So today I'm going to talk about some experiment that we run at UC Irvine. In our experimental setup, we had 2 subjects, each one with a head, EEG cap, and they were performing motor coordination tasks. So they were trying to perform some motor coordination, some joint motor coordination patterns. So in one pattern, each session they did one of the, each pair of subjects did 2 sessions. One session they did the synchronized pattern, which basically they are trying to synchronize finger tapping in a pad that they have, each one has. So, and they are like in cabins that they cannot see each other. They only see the feedback that they can see in the screen. So they are trying to coordinate the patterns. And the other pattern is a syncopated pattern. So in this pattern, instead of synchronizing, they are trying to tap between taps of each other. So, and as I said, we have some our trial structure here. Uncoupled trial. We have 3 types of trials. The first one is uncoupled trial, when they cannot, like, they don't have any feedback from each other. They only have their own feedback. So basically, they don't have any information of what the other subject is doing. And the second type of trial is a leader-follower. So just one subject have the feedback from the other, and of course we can have also the other direction. So in this type of trial we have twice the number of the uncoupled trial and the mutual trial that I'm talking about. The mutual trials both can see each other's tapping patterns. So we can think of as going from uncoupled trial to leader-follower and mutual trials as increasing the coupling between the subjects. Oops, sorry. Okay. There's a lot of spoilers there. Okay. So, and the idea of the analysis, the idea of our analysis is based on this idea of microstates. So basically in EEG, we have these states that are stable for a short period of time. Here we have an example. So basically the topographies of the EEG, of our EEG, they have like these stable patterns that they survive for a very short time. And we are basically trying to mimic, use this structure to create symbols and look at the dynamics of those symbols. So basically, we are going to discretize the EEG states that we have, and use this discretization to this symbol structure to analyze the dynamics. And okay, so how we determine those symbols? first we start with our EEG signals. So we divide the EEG signals in small time windows. Here, we choose time windows that we want to capture some dynamics that's not related to exactly the motor coordination, but we want to capture more than a single cycle of the coordination pattern. So these time windows are a bit large. They are 2 seconds, but they have an overlap of 50%. So it's 1 second of overlap. So so from these time windows, we have for each time window we calculate. We run a bunch of pre-processing steps on EEG on the EEG signals, and we calculate also the analytic signals. So we calculate the Hilbert transform, and we get like signals for each time window that are complex signals. So we have like complex values for each time point. And so for those that are familiar with analytical signals, they actually encode the phase relationship between the signals. And from there, we calculate the correlation matrices from these complex signals. So it's a complex correlation which captures some information of phase differences between the signals, and also the maximum amount of correlation that you can have from them. And okay, so from these correlation matrices is from where we go to get some discretized states. So from these correlation matrices, we use a measure to calculate a distance between matrices, and from this distance, we are going to coarse grain these correlation patterns. Okay, to do that, we do like an embedding, and then a Gaussian mixture model to cluster these things. And after that, we get like these sets of discrete states for each subject, and we combine them so we can have like this time series of joint brain states. So we coarse-grained these correlation matrices in sets of dyadic brain states, discretized dyadic brain states. Okay, so as I said, okay, so what is the distribution of those symbols? So here we have like some distributions, some numbers on what the amount of symbols that we found in each trial and each kind of session. So the important thing that we can extract from these numbers is first, most states are present in all 3 conditions. And second is that a very few states overlap between the uncoupled and mutual conditions. So it indicates that we have this difference in uncoupled and mutual conditions, and also, but the thing is, by the states, only the states themselves, it's very hard to distinguish the trial conditions. Okay, okay, but as I said, we are interested here in looking at the dynamics of the states. So basically, we are going to use 2 approaches here. The first one is it's a reference plot. So basically, when we visit the same states, it's a matrix that when we visit the same states, we put a 1, and it's a 0 when we are in different states. So it basically tells us, it gives us a measure of how stable are those states. And one nice thing here is that from these reference plots of this symbol sequence, we can see exactly each trial here, very defined in the reference plot. And the other approach is, well, what everybody here is looking for is the networks. So here we are using the networks in a different way that we usually use to analyze brain data. So instead of having a network of a given state, we have a network of those states on time. So we're basically trying to look at a network that encodes the dynamics of the brain states that we observe there. And remember that these brain states are not like a single brain state. They are like dyadic brain states. So it's a state between both subjects. Okay, so, so let's talk more deeply what is the recurrence quantification analysis. So as I said, when you have the dyadic sequence, the brain state sequence here, the recurrence plot is a plot, it's a matrix that has a 1 when you have like the same state, and for example, for a time I and a time j, if I have the same brain state, I put a 1 in the matrix. If I don't have, if they are in the different brain states, they are 0. And this way we can have like patterns like this. And what happens is that, in these patterns, vertical and horizontal lines, they indicate stationary states. So it's a state that they get there and they stay there for some time. And we can also have diagonal lines that indicates that we have a sequence of states that are repeating during the trial. And we analyze these patterns for each trial. And also, if we have like single points like here, it's when you have your dyad revisits that state that it visited at some point in time again. So every time, so basically it's telling us how often the dyad is recurring in the same states. And yeah, and also patterns from in this plot can reveal sequences and hidden periodicities. So it's basically a way to look at the phase portrait by having the data instead of like writing the differential equations and do all the analysis that we do for dynamical systems. Okay, so from what results that we can get from this kind of analysis? So what happens when we look at the relative average dwell time for this task? The average dwell time is basically I'm using relative because we are calculating the dwell times relative to the average of that dyad. Because each dyad, the individual variabilities are very large. So what is a long dwell time for one dyad, it's not the same thing it's not long for another dyad. So they vary widely between subjects. And yeah, so our brains are very good at capturing how different we are. So that comes from this fact. And what happens is that in the synchronization, as we increase the coupling condition that we have, we go from uncoupled to leader follower and mutual, we can see that it increases the dwell time. So the states tend to be more stable as we increase the coupling between dyads. And on the other hand, for syncopation, it decreases. So the symbols that we have there, they become less stable. And yeah, as I said, longer dwell times indicate greater stability in of the joint brain states. Okay, and the other thing is motif length. So the average, the length of the diagonal lines that we see in the recurrence quantification analysis. So it's basically how often a sequence of states repeat. And also, when the feedback increases, the motif length increases for synchronization patterns. and it decreases for syncopation. And yeah, as I said, the motif lengths, they indicate how when long sequences of states repeat more often. And okay, so and after that, the next analysis that we run on this within this framework is building the transition networks analysis. So basically, we are going to build a network more like the networks that Peter showed today for chemical reactions, but not here for brain states. They are like directed networks of the states of this dyad, of the brain states of this dyad. So what we are going to build is like a network like this, that we have like each node is a pair of correlation matrices, and this pair of correlation matrices, it gives us the information where, from a given pair of correlation matrices, where are the other states that we can go. And yeah, so these networks are they are based on the transition probabilities. So we build these networks from the probabilities that we can get from the sequence of states, and the links are also associated to transition probabilities. The links are we define weights for the links as minus the logarithm of p. It's related to the information content of that transition. So how if I have a transition that is very unlikely to happen, this weight tends to be higher. And if that transition that always happen has a weight 0. So basically, it says if I am in a given state, what are the it kind of relates the probability to go from another state to a kind of a distance. And okay, so in that sense, we can calculate the shortest path length. So it means that what are the sequence of states that are more likely to happen. And yeah, so when we compare the trials, the different trials, both coordination patterns feedback increase. When we increase the feedback, it increases the shorter path lengths. So we have like a set of states that the dyads go over more often, and when we increase the feedback, and this increased path length implies a more distributed flow along the network. So we have these less states that are visited very rarely, and a higher diversity of paths occurs for a given pair of nodes when we have this increased feedback. And yeah, and between centrality is a measure of how important, like how many nodes that we have that are part of this shortest path lengths. So it's basically a measure of how our network has like these central nodes that routes all the transitions that we have there during the task. And what happens here is that the average between the centrality decreases for the syncopation, but when we increase the feedback, but it basically, it says that we have a core periphery structure there in the network that's preserved, and for synchronization, because the between the centrality it's kept the same basically for synchronization as we increase the feedback. Yeah, so basically what these results tells us that the flow that happens during a syncopation task is more distributed along the network, while for synchronization, we have this core periphery structure that we have like a very important core that basically routes all the flow in that network. Yeah, so here the take-home message that we have from here is that the interaction conditions affect the dynamics of this motor coordination task. Different regimes uncoupled leader follow and influence the stability in the way that in the different ways for synchronization or for syncopation. So for synchronization, increased feedback lead to more stable brain states, joint brain states. And for synchronization, increased feedback reduces this instability. And from the network topology insights, we see that the synchronization, when you have synchronization, the synchronization pattern, the increase in the feedback keeps the core periphery network structure while it increases the path length. And for syncopation, stronger couplet led to a more distributed flow with longer path lengths and less traffic through the network core node. So it's a more distributed around the edges of the network. And yeah, and the idea of this work is like to study and identify basins of stability directly from the data, and yeah, so that's what I have, and open for questions.
Some questions? I have a question. When you show your the network of the transition probabilities, when
This?
yeah, if you don't have an edge, it means that the probability is 0 from one state to the other.
When I don't have what?
When you, in the case you don't have an edge between two nodes, because you have some nodes.
It means that transition never occurs in the
never
data.
occur.
Yeah.
This transition never occur, and I saw that it's somehow sparse, your network. So there are a lot of transitions that.
Yes. So these networks, they are created for each trial. So each trial is like a 2-minute trial. So it may be that it doesn't have time to occur yet.
Okay.
But so what happens is that in 2 minutes trials, we have around 80 to 100 symbols. So yeah, what happens is that in each trial we have so for each session we have around like 80 symbols for the dyad, but for each trial it keeps like in a very like concentrated region of the network, in which it visits usually like 10 to 8 edges, 8 to 10 states, depending on the trial. Some trials they have more, and yeah. So basically, what happens is that during the task, you have this kind of drift along the net. You can build like this entire session network, and that was the network that I show in the video. So if you are very patient and watch the video for a long time, you will see that it has a drift in the network. So it starts in one region, and after, yeah. So this is the entire session, right? So this is the network for the entire session. And the marked ones are where it is. So it starts like in a leader follower trial, and yeah, if we wait after some time, so it stays like in this region here, starts in this region, and after a long time. So this is a 5-minute video. You can see that it's in a very different region of the network. So it indicates that besides the effects that we are seeing from feedback and trial conditions, we also can see some other kind of effect, probably from like fatigue, or they are getting bored, or something else. So Yeah, that's a very interesting like sub-result that that we don't mention much, but I think it's very interesting because yeah, it's just because the brain is not like the it's not stationary all the time. So if you look at it for a long time, you see that it will change.
I have a question, but
Yes.
maybe again is a naive question. But I have worked in a very naive point of view with these microstates with a student, and one of the hardest questions that we have to deal with is how to choose the number of states. And
Yeah.
in some sense, we realized that, okay, maybe a lot of states would be a mess.
Yes.
And then we stayed like between 5 and 8. And it seems to me that you have a lot of states. So my question is, how do you choose the number of states?
Yeah, yes.
And well, you have a very sparse graph because you have a lot of states, right? So or not?
Yeah, it's around, as I said, it's around 100. But the thing is, we have this drift. So each session we have around 100, but it depends. As I said, it depends on the diet. So each diet we have like a different set of states and a different number of states. So it ranges from 80 to 100, and this. But the thing is, for each trial, you keep like in a set of like.
So this is
around 20.
maybe I have to do the question properly. So
Yeah, I will
how your criterion to
I-
choose the number of states?
I will answer your question. Let me just go back here. So yeah, so what we do is like we do this embedding, and after we do this embedding, we run a Gaussian mixture model. So when for a Gaussian mixture model, we can have like your Bayesian information criteria, and we choose the number of states that minimizes that. So yeah. So, and this is done for each subject. So for each subject, we have like around 8, 10 states. And then when we combine, we can have like this 80 to 100. We have a lot because we have 2 people.
Makes sense.
Dwell time is how much time it stays in a given state, in a given joint state. So it's like once we get there, how much time we spend there.
But this is already combined, right?
Yes, this is already combined.
Diet,
Yes,
right?
yes, this is the joint state of the diet. So it's like a state of the 2 brains. It's a combination of the 2 brains.
Hey, I was just wondering the first result that you showed, I think, on that RQA analysis. Yeah, the one after this slide, yeah. So here, yeah, I mean, you showed that like the synchronization increases with the kind of uncoupled, yeah.
Yeah.
I mean, the other thing that is more obvious to me just looking at the graph is that in the synchronization case, the variance is increasing.
Yes, yes.
What does that mean? Yeah.
So each of these points, it is like one trial, and it means that we have these trials that we have like a very very large dwell time, but even like in the same trial we have like a few examples that we don't have such a high value, but we tend to have, but not always. So again, there is a lot of factors going on there. It's a long experiment. They are like almost 1 hour tapping fingers. It's boring.
More questions? No, okay, let's thank again Ítalo. Thank you very much. So we have a break for lunch now, and we continue at 2 o'clock. Thank you. Okay, we will... ¿Funcionando? Sí. We will continue our session of the workshop with Andressa Cerqueira from University of São Carlos, and she's going to talk about recovering communities in networks. Thank you, Andressa.
Good afternoon, everyone. So first, I would like to thank you, Florencia, for the invitation. So IME USP was my home for 10 years. So I'm very glad to come back here. And actually, just a fun fact, the first random graphs in the brain, I was in my first year as a PhD student. So it's really good to be here now as a professor. So today I'm going to talk about recovery community networks, and I will show some applications in neuroscience. So, to start, I think everyone is familiar with networks. So I will start with this idea of recovery communities, so community detection. So we can think that I have a network, and that in this network, the nodes of the network can be clustered in different groups. That means that I have groups of nodes in such a way that inside a group, I have more connections between nodes of different groups. So this is the idea of a community. So suppose that I have this network with 3 groups. If I draw here, the adjacency matrix of this network, I just order the nodes of this network by groups, I can see this block X structure. So I have a lot of connections between nodes in the same, more connections between nodes in the same community, and fewer connections between nodes in different communities. So here, just the black dot means 1, and the white dot means like 0 in the adjacency matrix. But what happens in real life? So in real life, when we collect data and we have a network, we have something like this. This is the adjacency matrix that we observe. And then we say, okay, I just need to plot the graph, and I will see the communities. If I plot the graph, I will have something like that, right? So in real life, we don't have this really good word that we can see the communities. So what happens is that we need to employ some statistical approach to recover the communities, right? This is what we call community detection. And this is an area in statistics that people give a lot of attention now. Every time when you go to archive, you'll see, every day you are going to see a new approach to do community detection in random networks. So it's a very, statisticians are very interested in this kind of problem. So I'm going to talk a little bit about this problem today. So here just for a motivation, because I'm going to work with a special network that is weighted networks. So what we do when we collect the data? For example, we can imagine that we have some kind of stimulus. We record the data, can be an fMRI data or EEG data, doesn't matter. We have the time series here. We process the signal with some kind of method, and then at the end, I have, for example, a correlation matrix, right? So in all these arrows, there's a lot of math and stats behind, right? You can choose the best approach that you want to employ in each of these rows. So at the end, what I have, for example, here, I have this correlation matrix. What we usually do is just fix some threshold and then convert this correlation matrix to a binary graph. And then I do whatever I want with this kind of graph, right? We can say that in all of these arrows here, we are losing information, right? I don't care too much about the other ones, but I will care a lot about this one. So suppose that I have this correlation matrix. I don't want to fix a threshold to have a binary graph. Instead of that, I will say, okay, I have the correlation, so I have the strength of that connection. So I have 2 nodes, and I know how is the strength of this connection. I would like to use this strength as my data, right? So what I'm going to use is what we call a weighted network. So I will have a weight for each edge of my graph. Okay. So this is just some motivation why I'm looking at weighted networks. So when we can use weighted networks, for example, when we are analyzing brain connectivity networks, as I said before, what we can do, for example, is to have this correlation matrix, and then apply a Fisher transform in these correlations. What happens when we apply a Fisher transformation is that the data would like to look like a Gaussian distribution. This is what we have when you apply this Fisher transformation on Spearman or Pearson correlations, right? So we can apply this Fisher transformation, and then I have a weighted network where the weights are given almost like Gaussian distribution random variables, right? Or, for example, in another areas, for example, if you're modeling transportation networks, instead of just modeling the route that we have between 2 points, you know how much people use that route. For example, in air transportation networks, we can have like the number of flights between a route, and then we can have this weighted network. For example, in collaboration networks, social networks, and so on. So now, because of this kind of interest in measure the weight of a connection, people are studying more and more weighted networks. And this is what we propose in this work that I'm going to talk more about it today. So I will show here some way to generate a random network with communities, right? Because what I'm going to do here, I'm interested in community detection problems. So what I'm going to show now is a way that I will generate a network that has weighted edges and also have the community structure. So this is a generalization what there is in the literature that they call the stochastic block model. I just call like weighted stochastic block model. I mean, people are just doing a log and a log generalizations of stochastic block model and give another name. So what we have here. So suppose that I have a network with any vertices, right? To each vertex, I will assign a community membership. So I have here C1, C2, Cn, the community membership of each node, right? So this will be ID random variables for me in such a way that the probability that node I belongs to community A is given by some probability pi A, right? And I will assume that in this network I have K communities. So here I have K probabilities, the probability for each node to belong to a community. So in this case, K here is known for us. So I know how much communities I have in the network, right? Given that community, I will generate the weights for each edge of the graph. So given that I know the community of node I and J, the weight of the edge between the nodes I and J is given here by a normal distribution such that the mean depends on the communities of the nodes, and the variance also depends on the community of the nodes, right? Why I'm using normal distribution here? Because I have this idea that, remember, we have that correlation matrix for EEG or fMRI, doesn't matter, and then I apply Fisher transformation, then I would like to model as a Gaussian distribution. So I just supposing here I have a Gaussian distribution. So the idea is that conditional on the communities, the strength of the connection between nodes I and J has distribution Gaussian, has a Gaussian distribution with the mean and variance depends on the communities, right? And I just set like 0 in the diagonal because I don't have like self loops. Okay, it's just a convention. So here, basically what I have is a matrix B of means, because I have means, for example, to community 1 to 1, community 1 to 2, community 1 to 3, and so on. So I have K to the power 2 means, and here I have, this is not a covariance matrix, okay? This is a matrix with covariance entries. So I have here also K times K variances for each, depends on the community assignment that I have. And here just have that, given the communities, the edges are independent in the network. So that makes things easier to us to write the model. So then W is just a symmetric matrix as well, right? We don't have directions in this model. So the goal here is that I observed a weighted network. So I observe the W, the matrix W, and then I would like to recover C, right? I would like to recover the communities. This is our goal in this talk. Okay, so as a statistician that really likes likelihood, what we are going to do is use a likelihood approach. So today André talked a little bit about using different approaches. For example, looking at the spectrum of the graph. Okay, a lot of people do community detection looking at the spectrum of the graph, but what I like to do as a statistician is look at the likelihood, right? What's the likelihood? It's just the joint distribution of all the edges of my graph. In our case, it's just the joint distribution of all the weights of the edges in my graph. So what happens here? If we write the likelihood, we have here the probability to observe a community assignment, so C, given pi, and then here I have the probability to observe the network W given the communities. And then I need to sum over all possible community assignments. This is to have the marginal distribution of the W. And the problem here is that when we work with this kind of likelihood, we cannot compute this because here we have the sum of all possible community assignments. So even the case that we have only 2 communities and, for example, 20 nodes, this is impossible to compute. This is a huge number of combinations, right? It's exponentially large. So we cannot compute this in practice because of this sum here, right? But still, we can work with the likelihood. So in this case, what we can do, what was proposed in this work here of Eicher and co-authors, they use a method based on the likelihood, expectation maximization algorithm, but based on a variational approximation of the likelihood. So this is the kind of algorithms that we can use in this case because we cannot deal with the likelihood. And the problem, not exactly a problem, right? So they have this algorithm, but there is no theoretical results if this algorithm works well in practice, right? But there is this algorithm, this is well known, we can apply for any kind of likelihood distributions that we have here. Okay, so what I'm going to show you, okay. So this is the likelihood, whoops. This is the likelihood if I consider that my random variables, of course, are the weights of each edge of the graph, right, W. Okay, so if this is not easy to compute, I will change my random variable, okay? This is the idea of our approach. I'm not going to look the likelihood of the random variable W. I will just change, I will define a new random variable, and then I will look at the likelihood of this new random variable. And what is this new random variable? I will do that. So suppose that I have any community assignments for the nodes, right? Anyone. So I will keep C here for the true one, that one that generates my data. This is the C. E is anyone. I just pick one, right? And then what I'm going to define? I will fix a node in the graph, that's I. I fix node I in the graph. I will fix one community, for example, community K, and then I will sum all the weights of the connections to the node I that belongs, the other vertex belongs to community K. This is what I'm going to see. So this is just the strength of connection of node I with other nodes in the network that belongs to community K, right? So what we have here, since W's are independent random variables, if I sum Gaussians, I know that this is a Gaussian distribution, right? So, but what happens here? So here, I'm not summing on the true community assignment. I'm using another one. So what happens? I'm trying to explain to this picture here. So suppose that I have the true community assignments, right? That's 1, 2, 2, 1, 1. And then I pick another one. That's 2, 2, 1, 2, 1, right? Let me see. Next one. Oops. Okay, it's not easy to see, but okay. So I'm look at the node 2, for example, and then I will pick just, for example, k equals to 1, right? So I need to sum all the entries that E is equal to 1. So this one and this one, right? This and this one. I need to sum here. But in fact, these two random variables have different means, because here in the true one comes from group 2, and this one comes from the first group. So I'm summing here two independent Gaussian random variables with different means, because the means depends on the community assignments, right? So what I'm doing here is that, like, I'm not summing the correct random variables that I know what is the distribution, because if I sum like this, I cannot say that, where is the definition of the model here? That the mean is BC1CJ. I cannot say that because I'm summing the wrong entries of the matrix. So what we have actually is kind of a mixture of the means and a mixture of the variance. And this mixture here is given by how far I am from the true communities. So I fix A, there is the mismatch that I have between this community assignment with the true one, and I need to try to write the mixture of the Gaussian, the means and the variance of the Gaussians that I should sum to have, a Gaussian distribution that I know exactly what's the distribution. So what we do here, okay, so this random variable, this new random variable, what I know is that for each, if I fix a node, for each community, they are independent, S are independent, but if I compare, if I take one node and another one, so if I take like S1, Si, and Sj, they are not independent. So they are not independent because I'm summing the same edge twice in each S. So, okay, we have this. Si and Sj are not independent. And if I define the number of mismatches that I have in the true community with the community that I fix, the I community, so actually here is not the mismatch, but actually the number of matches, I can define the proportion of the matches that I have between the two communities assignments, and basically what it's possible to prove is not very difficult, but what we can prove is that this Sik that I define is a Gaussian distribution, because we are summing Gaussian independent random variables, that's okay, but the mean and the variance is different. The mean and the variance depends on the match that I have between the two communities assignments that I'm considering here. So now I have a new variance, a new mean that I call PLK, and a new variance that I call lambda LK. That's just a mixture of the means and a mixture of the variance that I have, all right? So now, instead of use W as my data, I'm going to use S, right? So now I have these random variables. Now I'm going to write the likelihood for these random variables, these S random variables. So if I write the joint distribution of Si, this is just a mixture, depends on the group of nodes Si. So conditional on node, the community of the true community of node I equals to L. I can write this probability here with the definition that I have here. This is Gaussian. So here I just have, since for different communities, these random variables are independent, this is just the product. And here we can plug in the density function of the Gaussian random variable with this mean and with this variance, right? But my data this is just for one node. This is just for node I. If I would like to look at all the nodes of the graph, I need to have the joint distribution of all S's. But what I told before is that the Si and Sj are not independent, right? But okay, I will look to the other side, right? And say, okay, let's see what happens if they are independent, right? If they're independent, I'm a really happy statistician because here I just put the product for all I, right? I equal to 1 up to n, and the product comes here, right? So this is what I'm going to do. So I just going to ignore that these variables are not independent, and I'm going to write what we call pseudo-likelihood, right? Because it's not exactly the likelihood of the model. I just pretend that everything is independent, so I have this pseudo-likelihood, and I'm going to work with this one. And I need to convince you at the end of this talk that it works well, right? So if I do that, I take the log of the likelihood, so the product that I have here, because I assume that the random variables are independent now, it's a sum, and here I have just a Gaussian mixture model. This really looks like a Gaussian mixture model. And then I can solve this problem using just a standard EM algorithm. I don't need to apply a variational approach to this EM algorithm. Just use the standard algorithm that we use when we have mixture of Gaussians. So here's the algorithm. Doesn't matter. I'm not going details in this algorithm, but what is important here is that I told you that we need to have some community assignment to start my algorithm, right? I need to fix A, then I need to run the algorithm, and then at the end, I have here the estimate of the communities. I had here C hat, right? The estimate of the okay, better, right? So we know that depends on the starting point, we can have different results, right? So I'm going to see, okay, how we should start this algorithm in order to have a good community, the good estimation for the communities here. Okay, so we need to answer this question, how the choice of the initial community assignment I affect the C hat, right? The estimation of the communities. Okay, so first, I need to do some math. We always need to do some assumptions, unfortunately, right? So first, I will assume that the initial labeling, I met C in a fraction of nodes gamma, okay? So I will match C 10% of the nodes. So 10% of the nodes match, the other one is in different communities, right? I will do that just because it's easier to just track this parameter gamma. Otherwise, it will be a mess to see what happens with the algorithm. I define the error, so this is just L C hat C is how many mistakes I made with my estimation with the true communities. This is just because we need to relabel, so it doesn't matter too much, but this is just the error that we would like to see if this error here goes to 0, right? In this case, we have a good algorithm to recover the communities. And of course, I need one more assumption just to make things easier to write. I will assume that the mean weight between each group, for example, if I take 2 nodes that belongs to the same group, the mean of the strength of the connection is mu1, and if the nodes belongs to different groups, is mu2. And now I just need to look at these 2 parameters. Otherwise, I need to take care of k-square parameters. That's too much, right? And I will fix the variance the same for all the communities. This is just to show a good version of the result. So in our work, that's going to appear in 2026 in a special issue, in a special issue for networks in Statistical Kanika. That's great because I can always use this work, for example, everywhere. But if you want to check out this work, it's on archive, so it's you can open and take a look. So what we prove, so this is, I did this work when I was a postdoc at the University of Michigan. So this is a joint work with Professor Lisa Levina for University of Michigan. And what we prove here is that, suppose that I have, so this is just telling us that, okay, suppose that I have a network with balanced communities. That means that the number of nodes in each community is the same, right? So what we prove? We prove that in one step of this algorithm, if I look at the error between the estimator, if I starting with configuration E, with the true communities, we prove this upper bound here for this expected value. What's this upper bound here? If you look, this upper bound is going to 0, so case fixed, right? Case fixed for me. So this upper bound is going to 0 if the difference between mu1, mu2, the difference between the means increases, right? And it also increases because it's the size of the network. So if the means of the groups are too close, it's very difficult to do community detection because I cannot separate the groups. If the means are different, become different, then it's easier and easier to recover the communities when the size of the network grows, right? So this is what we have for the upper bound for the error. So this error, when n is going to infinity, is going to 0, right? And what is interesting is that in 2020, in this work, Xu and co-authors proved a lower bound for any cluster algorithm for weighted networks, right? And the lower bound that they got for the expectation of the error, if you see, is almost the same, our upper bound, up to a constant, right? Because here is just n, the difference of the means, the variance, and here we have is the same quantity here. So up to a constant, our upper bound matches the lower bound. That means that our algorithm is as good as possible, right? And we can also make, for example, the means and the variance also growing with n. Doesn't matter what, the only thing that we need to have is that this quantity here goes to infinity, to have that the upper bound goes to 0, right? So we can also have the means and the variance going to growing or decreasing with n. Okay, so I bring here some simulations, but we have more simulations in the paper. Actually, in the paper, we have other proofs for general cases, for example, for unbalanced network. But since the bound is so too ugly to show here, just keep it in the paper, right? So if you want to take a look at that, everything is there. So I just bring some simulations that we have done here. So suppose that I have 3 communities, right? This is the matrix of means. This is the matrix of variance. I just fix the variance, and I vary here the difference of the means, right? So x is here is just the difference of the means, and the y-axis here is just the error. So I want the error going to 0, right? So for balanced network with 500 nodes, I compare here several methods that I'm going to explain now. Let me see. Okay. This orange one, SC, is a spectral cluster, right? It's a method based on the spectrum of the graph. They don't look at the likelihood of the graph. So I just apply spectral cluster. So it's this orange line here. And the green line, what I did, just take the result of spectral cluster and use as the initial input of my algorithm, right? And if you see, there is a slight improvement here. Okay, I'm sorry, the green line's not so easy to see, but we improve a little bit, right? The spectral cluster. The other methods, let me see. The red line here is the discretization method that was, let me just come back here, is the method that they also propose in this work here. They prove the lower bound and also propose some methods to achieve this lower bound. And I apply the method that they propose, is this red line here. And then if I take the output of their algorithm and use as input of mine, you can see that in this case I improve more, right, the result of this measure. And the purple one it's just that I just start, I mean, this is simulation, so I know I know what's the true communities, right? So I just flip 30% of 70% of them, and then apply my algorithm. This is why purple is really good, because I'm cheating here in some case, right? And you see that when you increase the difference of the means, their error goes to 0, right? This is for balanced communities. For this is for more nodes, so just to see that, yes, goes to 0 when we increase the network. For unbalanced communities, so here I have 20% of nodes in community 1, 30% of nodes in community 2, and in community 3, and 50% of the nodes in community 2, right? So I have unbalanced communities. We know that spectral cluster in this case can fail a little bit, right? Because of you have this kind of unbalanced number of nodes in each community. So we can see that our method can improve now more the spectral cluster and the other ones. So this is interesting because we can use another algorithm as an initialization of our, and then we can improve the result. So this is a way to choose E at the beginning of the algorithm, right? I can make it random, or I just use the output of other method, right? Which one, what? The one is the purple. Oops. It's this one. But okay. So in the paper, we have more simulations. For example, I also run simulations for, here I just sample for Gaussians, right? So in the paper, I sample for example, mixture of Gaussians, T of students, and then the method works well, right? So this is to show that, okay, maybe also, for example, if we also have mixture of Gaussians or distribution with a heavier tail, the method also works, right? And for application, we apply it for community detection when we have fMRI data. So now I need to I need to apologize because now is a statistician talking about this biology thing. Okay, so forgive me if I make any mistake here. So what we have in this data, we have a data of resting state fMRI images of 54 individuals with schizophrenia and 69 healthy patients, right? So here I have a sample of network, but my method works when I have one network. So the first thing that I did here is just to combine the 54 networks together to have only one. So I just apply some kind of method that was proposed for Kit Levine for example, take an average of network, let's say like this, and then I combine a lot of networks in just one. And then I use the same method to combine the 69 health networks in just one. So here, just think that I have two networks. One with individuals with some condition, and another one with health individuals. So here we have a network with 264 nodes. And I did exactly what I explained before. Use correlation to compute this correlation matrix, and then after that, I apply the Fisher transformation for the correlations, and then I use these for the weights of my edges, right? So since it's difficult to say what's true here, right? I try to compare the results with some parcellation that's proposed in the literature. So here, I'm sorry that I forgot to put the work here, but I use this power parcellation. That's just a way to divide the nodes of the regions of the brain in some functional areas, let's say, like this. This is a statistician trying to explain this kind of things, right? So I use this power parcellation, this kind of division of this node. We have 14 groups. So I fix here k equals to 14, right? In this case, I don't need to estimate how many groups I have in the network because I would like to compare the clusters that I'm going to obtain with these regions, right? So this is what I'm going to fix as grand truth, right? So I estimate the communities. I do community detection using our algorithm. So what I did is just using spectral cluster, has initialization, and then I apply my algorithm to the output of the spectral cluster, and I fix k equals to 14. And then I'm going to explain what this colorful picture here, right? So let's think that here, H is the regions that I estimate for the healthy network, right? And P here is the regions of the power parcellation. So this quantity here that we can show here in this flow is how many nodes are in this first community for the health that belongs to the first power parcellation, right? So this flow here show how many nodes belongs to these parcellations here, right? I don't know if it's clear. This is the best way that I find to show the results, right? And here we have the community estimated for the health network and here for the schizophrenia, right? So when you look at this picture here, we can see kind of homogeneous thing happens. For example, all the nodes here are in this community, right? So I can say that this region behaves at the same as this region here, right? So when you look at this side, the right side, we can see that a lot of nodes goes to the same community here, right? But there is this H11 here that we can see that splits a little bit. H2 as well, it splits a little bit. And what's interesting is that if we look at H11 here, most of the nodes in health is going to power parcellation 5. H2 is the same, most of the nodes is going to power parcellation 5. The same, for example, if we look H6 here, it splits a lot, and most of the nodes going to power parcellation 5. And when you go back, this power parcellation is related with the full mode. As far as is known to be related, this region is known to be related with schizophrenia. So this was an interesting result that we got, right? Because we found something that's in some sense null, so it seems that the method for community detection is working well, right? Because we can recover something that was expected in some sense. All right. So what's interesting, so we can see that in theory, the method seems to work because we prove consistency. In practice, the method seems to work through simulations and through the real data. But there are a few things that we can extend when we analyze this kind of weighted networks. That is, for example, in this case, all the edges has a strength, right? But sometimes we can have, for example, no connection. I don't know if the brain, if happens, so I don't know, so I'm going to say something in transplantations, for example. In airline transplantation, we can, for example, there's some airport that there's no flight to another one. So this edge doesn't exist, it's zero, right? And in this way, the way that we model here, we are going to model that, oh, this edge exists, but the strength of it is zero, right? But another way to model it is just first throw a coin to see if the edge exists or not. And then after that, you put an edge on that. In this case, we are able to model sparsity in the network because I can model like zeros in the network. So this is what I put here, that we can incorporate edge distribution with a point mass at 0, right? If you are interested on that, my master student and I, we just put on paper on archive that we do that. So we consider this kind of models using a zero inflated Poisson distributions. So there is this kinds of zero inflated distribution in stats that you can model data with a lot of zeros. So in this case, we are able to model sparsity in a weighted network. Right? So, but I think there's more that we could explore and do in this direction. And that's all. Thank you.
Okay, thank you very much, Andressa. Some questions? You have some questions? Here, okay.
Okay, thank you very much for your presentation. First of all, you said that you have a pseudo likelihood. Why not you try to derive a kind of pseudo AIC to estimate the number of communities?
Yeah. Actually, I didn't thought too much about estimate the number of communities for this problem. It's open, but it's possible to do something like this. For example, since the pseudo likelihood that I have is just a mixture of Gaussians, we can use some results for a mixture of Gaussians
exactly,
to recover K. Yeah.
Yes.
That's right, yeah.
Okay, and my second question is, in the last part of your presentation, you compared, I think, two data sets, the clustering structure, yes, this one. You have two data sets, I think. I don't know, I don't remember, 100 people and 100 people, right? And you clustered the brain of group A and clustered the group B, and you said that you have, how can I say, the items of each cluster changed. I mean, the clustering, right?
Yes.
Clustering structure.
Yeah, the nodes, yeah.
Yes, the nodes.
It's changing for communities, yeah.
Yes, and you are showing here a kind of average, right? Between two.
Yes, exactly. I'm showing the average here.
But indeed, you have many replicates, right?
Exactly. I didn't apply the method for any patients, yeah.
Yes.
But I could, yeah.
Yes, but probably you calculate a kind of average, right? And apply to your community detection on this average, right?
Yes.
Yes, okay. Why don't you test if they are, in fact, changing the clustering structure?
To each individual, you mean?
No, for the average, like a t-test or an analysis of arrays, but for clustering. Because you could, not only visually, but you could test, in fact, if the clustering structure is changing, in fact, between that. And also, which item is changing.
I see. You mean like to test this configuration with this one, right?
Yeah,
this cluster
exactly,
with this one.
Yes, yes.
Yeah.
Because here you have only, you can, well, visually see it.
Yes, exactly. Just a guess, yeah.
Yes. Okay.
Yeah, it's interesting. Thank you.
Thank you.
Another.
Thanks for the presentation. I would just like to understand this a little better. This H1 to H14 is sets of nodes?
Yes, it's the estimated community for each node. So here I have all the nodes that are estimated to belong to community 1, and so on.
Okay. And you are comparing this with the power parcellation?
Exactly.
Yeah, so what you want to see is if the communities do match with what is...
Exactly, because I don't have the run through here. So at the end, I will estimate the community, say, hey, this is the communities, and yeah, that's it. So I try to compare with some known division of the regions of the brain in functional areas, right? To compare what happens with healthy patients with the patients with some condition.
Okay, and in the right side, you have schizophrenic patients.
Yes.
Okay, so basically what you are showing is the difference between communities established between these health people and...
Exactly,
okay. So,
yes.
Yes, thank you.
Another question? Okay.
It's a very simple one. You suppose that actually your intra and extra mean of interaction are more or less the same,
Just
right?
for the proof.
Okay. This
Yeah,
was
the
my question.
algorithm. Yeah, this is a good question because the algorithm works well for.
Yeah,
you don't
because
need
you're
to have
just
this assumption.
comparing the intra and extra in your
Yes.
estimator, but then I suppose you have a maximum or a sum in.
I could have something
in
like
the
that, yeah.
communities, right?
Yes, if I was not lazy with the math, I could have, yeah.
No, it's not lazy. It's just because the notation is a mess. But
No, the notation, yeah.
But for the simulation, so you are in the.
Yeah, I don't need to have an assumption.
Nice.
I was trying to think about the difference of your approach with clustering in general. And I think, let me see if I thought, okay, it's okay what I'm thinking. You have only, when you have the strength of the edges, it's like you have
A similarity.
a similarity.
Exactly.
But if you're thinking on the inverse of this similarity, you can think as a distance, right? That is something that you use in clustering. Because when you have vectors, you use in general the distance between the points to do the clustering. But so only a comment about this, that I think the difference, and one thing that is very nice of your work, is that you don't need to have the location of the points, because to do clustering, if you want to use something like k-means, you need to have the locations because you need to compute the means, not only the distance. And you are doing a clustering having only the distance. It's like, suppose you have only some distance between points.
Yeah, we can think like the weighted network has a similarity matrix or something like that, because we know how close, how strong is this connection, right?
But you have less information than in clustering, because in
because
clustering
you have the positions.
Yeah.
I see what you mean. Yeah.
So I only thinking about this, that I think that is one strength of your work, that you can use this clustering only when you have some partial information like the distance or the similarity, and not the full position of the point.
Yeah, that's true. Yeah.
Okay, maybe.
Yeah, I can change. I mean, maybe until 2026, I can change something in the paper.
Okay, only this comment. Another question? No?
Thank you, Andressa. Very clear and very nice presentation. My question goes along, I think the one Paulo asked. Is it possible to estimate the performance for the individuals, for instance, for the patients?
Yeah, I could apply the same method for each individual, but it was a lot, and it's difficult to take track what's going on, because I have students working on that, and each individual behaves a different way. And then when you have the communities, they're all different, and then you look at a lot of community estimates, say, okay, what I have now, how I will combine this information to get a result, to have an interpretation. So this is why I put everyone together, just to have one network. But yes, it's possible. I can apply the same method for each individual.
Yes, if you have a scale, for instance, of the gravity of the symptoms, things like that, and you could maybe try to
add this to the model,
link.
yeah.
Yes, yes.
This is interesting.
Okay, thank you. Let's.
This is a small one too. In your simulation, when you don't use any other algorithm to use as input of your algorithm, you told us that actually you take the true one and then you flip 70%.
Yes.
Why 70%? Why you just don't choose randomly everyone?
No, I did a lot of things. For this picture, I just.
But it doesn't matter, right? The only thing
matter.
if
yeah, because at the limit, gamma doesn't matter. It's a constant.
I mean, if you just choose randomly, the only difference would be that you probably would
take more time.
Take more time. Okay.
Yes, because if you see gamma is here, so doesn't matter too much when n goes to infinity.
Okay, I think there is no more questions. So let's thank Andressa again.
Thank you. Thank you.
So we have 5 minutes if everyone wants to. No, there is no coffee, but is that? Yeah, it's an online. So we are going to have Okay, maybe we can begin. Maybe, okay, if she's there.
I'm here. Can you hear me?
Hi, can you hear us?
Hi. I can hear you. Can you hear me or see me?
Yes, I can see you
Okay.
and I can hear you.
I don't see the audience anymore. So is there a way
Oh.
to make it happen?
I'm not. No, it's not possible. Sorry.
Oh,
I just
okay.
discovered too. I'm sorry.
Okay.
But
It's okay. So they can see me. Is it like am I?
yeah.
Okay.
Okay. So if you want to share your slides, just to check
Yeah.
that
Let me.
we can see it.
Sure. Yeah. Just give me 1 second. Do you see?
Yes.
And still like with the presentation mode on and everything, it's good?
Yes.
Okay, perfect. So should I start? I don't see
I'm
any.
checking.
Yeah.
1 second.
Okay.
Can you start? So maybe we should start. We are 5 minutes in advance, but I don't think it's a problem.
Okay.
Okay. So I will present
right.
you very quickly. So our next speak is Kanika Bansai. Is it right? Your
Yes,
pronunciation?
perfect.
Good.
Yeah.
So thank you for accepting our invitation and feel free to start.
Yeah. Thank you. Hello, everyone. I don't see anything or anybody from there or myself, so I'm kind of like blind here. Hopefully everything will work out well. And thanks to Claudia and her team and Ítalo for having me present at this workshop. It has been great so far. And I know I'm the last one, so probably everyone is tired, so I'll try to keep it quick and light. And I'm gonna discuss about a lot of experimental data and a variety of neuroimaging data and some behavior. And I'm super glad that I'm speaking after Andressa because her presentation made my life really, really easy, and you'll know why. But let me just begin with a little bit of my background. So I'm based in New York. I'm a senior fellow at the US Army DEFCON Army Research Laboratory. We do a variety of basic science research. And I'm also an research assistant professor at the University of Maryland, Baltimore County. All right, so let's dive right into brain flexibility. That is what I'll be discussing. And when we think about brain flexibility, intuitively, it kind of makes sense. It kind of maybe makes us think about how well we switch tasks or adapt to different environmental changes or grow or learn and a lot of different things. So to catch that common perception, I took a shortcut and I asked ChatGPT to give me a cool title around brain network flexibility, and it's called Cognitive Implications. And it gave me a bunch. I'm highlighting some here. And what I wanted to say is that these majority of these titles, like here or here, they associate flexibility with some sort of positive trait or power in a very qualitative way, of course. And through my work, I would like to convince you that flexibility is something that allows or gives us cognitive resilience or adaptability. So it's a good thing in a more quantitative way. And of course, going with the theme of the workshop, I'll be taking a networks perspective. So we'll start with quantitatively defining what I mean by flexibility or network flexibility. And then we'll walk through some computational challenges of implementing how to extract flexibility from from brain data. And then I'll discuss some examples of how flexibility plays a role in cognition and like agility of the brain. And then I will briefly discuss about how do we harness flexibility or modulate it. And then I'll end with what is flexibility beyond the network perspective. All right, so I think I don't need to convince this set of audience about why it is a good idea to study brain from a complex networks perspective. Brain is a network of networks. There are a lot of interactions happening, and a cognitive emerges out of those interactions. So like taking a network perspective to understand cognition is sort of makes sense. It's kind of intuitive. And there's another beauty of networks approaches is that it can be applied across different scales. So for example, I could use similar methods and frameworks at neuronal level or neuronal population level or at larger brain level where there's a lot of complexity and accuracy is sort of lower in terms of like detailing of, in terms of biological details. And this is where my work kind of lies. And then there's another aspect of brain networks that's structural, and also there are functional networks. So structural are the ones which tell us how different regions or nodes are physically connected or wired. These are usually extracted through white matter bundles or they're called connectomes. And the other way is, I think a lot of you talked about functional connectivity today, which is some sort of statistical or functional relationship between different brain regions or nodes. I think the simplest one is correlation that we just heard about in the previous talk. There could be more nonlinear or more sophisticated ways to extract this functional relationship. All right, so across multiple studies and years, some properties of brain networks have been established. For example, we know that brain networks are not random or regular. They are somewhere in between, which is defined by the small world architecture. And it says that there is high clustering, meaning neighboring nodes have cliquish connectivity, which makes it really easy to communicate within your neighborhood. But at the same time, if distant or physically distant nodes had to be connected or needed to communicate, it's also easier to do that because there are long range connections available as well. The other one is this core periphery organization where there's a strongly connected core, which is highly dense within itself, and there are some peripheral nodes. So this also helps with communication in the brain. And then, of course, my favorite one, and what Andressa has been talking about as well, is the modular architecture, where the brain is divided into different modules or communities, which are these clusters of highly connected regions, multiple clusters of highly connected regions, which have low connectivity in between, some connectivity, but low connectivity. And it turns out this modular architecture is really important for brain functioning. And there have been a lot of papers, they talk about how these modular architecture changes with age, showing some sort of aging related processes or development or learning across lifespan. And also how this modular architecture changes with task demands. So when different tasks are being performed, different modules appear, they reconfigure, they kind of disappear, and so on. So a functional definition of flexibility comes from reconfiguration of modules. And I'm gonna talk about this kind of milestone paper from Dani Bassett and her team. And what they did was they studied a motor learning. Particularly, they made participants play some piano notes, a piece of notes, using their not dominant hand. And then they tracked how they were learning that using, like pressing those notes over time. And they found that during learning process, brain communities or modules, they kind of reconfigured. And then they extracted how flexibly different nodes within the brain made allegiances to these modules or communities. And they found that motor learning was better for individuals who had better flexibility. So this idea of flexibility was originated here, and also its association with cognitive abilities. So I'm going to use this kind of framework or this definition of flexibility in my work. All right, so first part is to actually detect these modules or communities. And as we heard just now, that it's a field of research in itself. There are a lot of new algorithms every day that kind of optimize how to detect certain kind of communities. There's always a debate of what does better, what doesn't do better, and all of that. But basically what we use is modularity maximization. It's Newman Girvan gave the idea many years ago, 2 decades almost. And it literally optimizes or maximizes this quantity, modularity, which says that there is more connectivity within modules than between modules. And there are different ways to kind of realize the algorithm, but this is the fundamental to it. And these are different quantities. Q is modularity. Here we have total sum of the edges, and here we are talking about weighted edges in the context of brain. This is the adjacency or connectivity matrix, degree nodes, and here these quantities represent community assignments of node I and J. Interestingly, here there's a resolution parameter which drives how you can detect how much of a smaller or larger communities can be detected. So this kind of puts a limit to what can be detected in a network. And to just give you a quick example, this is, I took this from Wikipedia. It's a very simplistic network. And if we were to apply modularity maximization here, we can clearly see network partitions into 3 communities where there are higher connectivity within modules and lower connectivity between modules. But brain networks are very complex. They are, of course, weighted. There's a lot going on. So it's not as trivial to extract these communities. And sometimes there's no ground truth. There's just like, it's more of a data-driven kind of approach. And then there is another problem of these community evolving with time. So functional brain evolves very rapidly, depending on which modality we are using for imaging the brain. It could be in milliseconds or seconds or even days. But there is evolution of these communities. So we have to keep that in mind. So for that, there's this generalization of community detection. It's called dynamic community detection, which takes care of detecting communities across multiple consecutive layers. And now we also have this additional resolution parameter called gamma, which dictates how well a node is connected to itself in the next or consecutive communities. So now we have these 2 resolution parameters to think about while extracting communities. Now assume that magically in some algorithm we do extract these different communities, and then we track how between different layers, different nodes change their community allegiance. For example, if we just look at this reference node here, it made a flip here from green community to yellow community. And then again, it made a flip here from yellow community to green community. So flexibility is defined by for a particular node is the number of swaps it makes divided by the total number of swaps possible, which is actually number of layers minus 1. And these swaps can happen in many different ways. It can be between, say, between two communities, back and forth. It could be that a node could be a part of more than two communities, multiple communities. So all of the swaps are kind of included. So it's a likelihood of a node to change its community allegiance. So if on an average, if nodes are more likely to change their community allegiance, the network is more flexible and so on. All right, so this kind of definition of flexibility, it has been already shown that it supports various cognitive functions. For example, I already discussed this motor learning with piano notes. There was another paper that followed after this. They showed that the higher flexibility in frontal cortices, frontal regions of the brain, the higher flexibility of those regions was related with better memory performance. And then there was another paper following this. They looked at a particular brain region, it's VMPFC. I forgot what VM was, but PFC is prefrontal cortex, which is very important in social neuroscience. And they found that the flexibility of this particular region was related to the likelihood of a person to take social messaging more effectively. Particularly, they were talking about smokers, and they concluded that if a person has more flexible VMPFC, they were more likely to quit smoking after some advertising or social messaging. Recently, in this study, a distributed set of brain regions were found to be directly related with creativity. And this very recent paper says, using a human connectome project data, that generally openness and general intelligence is related with the flexibility of the brain. Us, over the years, we have also extended.
Kanika? Are you there? I don't think so. Oh. Caribe? Do we have news? Nossa, mas foi muito estranho porque caiu. Ela saiu?
Então, eu não tenho certeza, eu estou tentando entrar de volta na mesma sala. Vocês têm uma comunicação direta com ela?
Estão falando lá com o computador.
Tá, fala para fazer mudar o link. Deixa eu só ver. Isso, mudar o link.
Hello.
Hey, sorry. I think I don't know if we lost connection, maybe?
Yeah, I think that's okay.
Can you show you more or less where you are, and then you tell you more or less where we fall?
Sure, yeah. Let me share my screen again. Okay. All right.
Yeah, I think it was there.
You're there? Okay, perfect. Okay.
Was
So
quick,
it
I
wasn't-
think.
Yeah, not much was lost. I heard some weird noise, so I
Sorry.
thought of checking in again. No worries at all. Now everything is good, right? You can hear me, see me. Yeah, all right. Okay, so I was talking about how through our work we kind of extended flexibility across different contexts, going beyond traditional laboratory kind of studies. But somewhere down the line, we kind of realized a slight problem with how we were kind of defining everything. So ideally, what we have is some sort of time series data, some sort of neuroimaging. It could be fMRI, EEG, or ECOG, or whatever. So there's some windowing or epoching that happens. And then in this windows, we calculate connectivity, network connectivity. And then we stack these into multiple layers, and that forms our dynamic or multi-layer network, and then calculate the community, dynamic community. And then from that, we extract flexibility as a measure for each individual. So we could analyze individual properties, group property, and properties, and whatnot. But the challenge is about these resolution parameters. So there was gamma, which was dictating how small or large communities can be detected within a layer. And also there is this omega, which dictates how strongly two nodes within, how strongly a node is connected to itself in consecutive layers. And it turns out, based on how we choose gamma and omega, the community structure changes widely. For example, if we start with low omega and low gamma, we could end up in every node in one community, and that community changing over time, kind of thing. And then if gamma is too high, then each node is its own community. If omega is too high, then all different layers are more similar in terms of community architecture. So that's a problem. And choosing a good operational value of gamma and omega is kind of hard, if we just try and optimize these parameters and look at what emerges and try to pick a value. So we thought we have to come up with some sort of optimization which is more objective, and Ítalo was leading the project. So to optimize the spatial resolution or gamma parameter, we looked at how community sizes change with time, right? So for example, here in the first layer, there are 3 communities of size 4, 3, and 2 nodes respectively. And in the second layer, they kind of change, and now the community sizes are 5, 1, and 3, and so on. So we looked at how the distribution of community sizes change with gamma. And it turns out that if gamma is high, there's a skewness towards low sizes of communities because nodes tend to separate into their own communities more often. And if gamma is too high, then it becomes more likely that too many nodes are in the same community, so community sizes kind of become as much as the system size itself. So then we decided that we need to pick a gamma such that the skewness of this curve, which says bias either for low community or high community, is the lowest. And this is just for an example data set. So this is where we would pick our gamma so that the skewness is the lowest. And to optimize the temporal resolution parameter, we looked at this temporal aspect where we extracted prevalence times, which is for how long or for how many layers of a network, a node stays within the same community. For example, this particular reference node is in community 1, and then it changes for just one time layer, and it changes to another community for 2 consecutive time layers, and so on. So now when we look at the probability distribution of these prevalence times, there are again some bias for either communities changing very quickly, or they're just staying for too long. And we propose that if we optimize these probability distribution to be close to a scale-free or a power law distribution, that would make sure that there is no scale bias or bias towards community changing too quickly or too slowly. So we kind of extracted these prevalence time distributions, fit a power law, and then pick a value of omega where the difference between a fitted power law or a synthetic power law was the least. And of course, we needed to validate the method, and we did a lot of validation with benchmark synthetic kind of networks. But I just want to quickly also tell about this real network, very complicated, but here we knew the ground truth. It was the contact network of ant colonies. And this was beautifully designed, meticulous setup where the experimenters were looking at 6 colonies for 41 days. There were like millions of interactions, and social contact was determined by physical vicinity of ants. And they used something very different, infomap-based community detection and visual inspection to detect communities. And this kind of summarizes their findings. They found 3 different communities of nurses ants, forager ants, and cleaner ants. But interestingly, 2 of those communities kind of showed up in their algorithm. But one of the communities, the cleaner ones, they found through visual inspection, they just observed that, okay, there's this 3rd community which interacts more often with the rest of 2, but they're kind of doing something else. And what they also found that these roles of different ants within the network also change with time. So we use this data, applied our community detection and optimization, and we could clearly see at any given point across days that there are 3 communities, and these communities kind of evolve with time. So I thought that was pretty straightforward without having to do any visual inspection and so on. All right, so let's just come back to brain and brain flexibility and its relation with cognitive abilities. So I'm going to give you this example first, where this study was about sleep and cognitive resilience. So it was done at UC Santa Barbara in collaboration with Army Research Laboratory, and about 50 to 60 individuals came to the lab for multiple sessions, for up to 8 sessions across 16 weeks. And they performed different cognitive tasks, very controlled laboratory kind of tasks related to vigilance or memory and attention. And their sleep records were also collected using an actigraphy watch. So we knew before they came to the session how much they slept the day before, and we also knew the longer term history of their sleep. So here I'm showing just the total sleep time just the day before the experiment was done. And we saw really good naturalistic variation. We did not put any artificial control over their sleep, which is usually done in sleep-related studies, but we just let it vary naturalistically. And even with that kind of naturalistic setup, we saw low sleepers, which are sleeping for less than 6 hours, and high sleepers, which are kind of having a good amount of sleep, and some people in between. But interestingly, what we saw, we were expecting that there would be some change in their task performance with change of sleep. But we did not find any. For example, this is performance on psychomotor vigilance task, which is very sensitive for sleepiness. And what you have to do is respond to a stimuli as quickly as you can as it appears on the screen. So performance is measured as response time. And across these sleep categories, less than 6 hours, 6 to 7, 7 to 8 hours, and greater than 8 hours, there was no statistical difference. We ran a lot of different statistical tests. And we also found this kind of same pattern across different tasks. The other one was working memory task, where the participant was shown a pattern of structures, and then after some time the pattern was repeated and they had to remember if it is same as before or not. And there was a modular math task where they had to respond if a mathematical expression is correct or not. But we did not see any sort of performance change with sleep categories, which was kind of interesting. It kind of tells about cognitive resilience of the brain. It says that there's the maintained performance irrespective of sleep across multiple tasks. So we tested if there is some change in flexibility while these tasks are being performed. And then we used ordinal regression, which tested the likelihood of flexibility changing while the task is being performed from one group to the other. That means one category of sleep to the other. And this is here I'm showing the coefficients of that regression across different functional components of the brain, which Andressa mentioned briefly in her talk. So these are different functional systems or communities predefined, which kind of combine different regions or nodes in the brain according to their cognitive role. For example, visual is pretty intuitive. It combines nodes which are involved in visual processing. So anyway, and so this inner circle represents the significance level. So what we can see is that there were significant changes in flexibility as the task was being performed, depending on which sleep categories the person was falling into. And in particular, it kind of said that as the sleep increases, or I would say as the sleep quantity decreases, there was higher flexibility of that network. And especially, we found that during resting state, which is a state of no particular task or mind wandering, where no particular cognitive process is kind of enhanced or targeted, there was no relationship between flexibility and sleep categories. And then we observed another thing. So apart from looking at the sleep of an individual the night before, we also looked at their longer term sleep quality. So we looked at sleep regularity index, which tells whether they sleep regularly at the same time for a regular amount of time every day or not. And we just did a loose median split of regular sleeper and irregular sleeper. And what we found is that the relationship between flexibility and sleep is more pronounced for irregular sleeper. So to kind of summarize this kind of complicated thing, we saw that task performance is maintained with low sleep, and there's a simultaneous increase in flexibility when the sleep is lower, and it is very task specific. It happens in different cognitive communities for different tasks, and it is absent in resting state. And we concluded that flexibility could provide a compensatory mechanism against these environmental fluctuations. So it makes the brain resilient, and it compensates for that lack of sleep that ideally should lead to lower performance. And clearly, long-term sleep quality moderates this relationship, which kind of makes sense. All right, so another example, it's related to adaptation of an exoskeleton boot. So in this one, individuals were wearing an exoskeleton boot, and they were walking. And the idea was that if they are using the exoskeleton well, they would be using their own muscles less and less. So as they were walking, their brain activity was monitored using EEG, and their muscle activity was also monitored to see how much they are adapting or how much well they are using the boot. And this was the study design. So they just started walking with the boot on, with putting the boots on. And during this experiment, boots were randomly on and off to kind of see while the how much of muscles are used while the boot is on or off. Then there was training when they walked with exoskeleton boot for hours to kind of get acclimatized to it. And there was a session where again they walked while their brain activity was monitored, and the similar setup was repeated for boots on and boot off. And this is what we observed. So this on y-axis I represent muscle change. So negative means there was less use of muscle while the boot was on. So negative is good in terms of adaptation. So negative means better adaptation. And here, this is pre-training condition, this is post-training condition. And it clearly we can see that post-training adaptation definitely became better. So with training, everybody kind of adapted to using the boot. But there was a huge variability in the beginning. And we can see that there were some individuals who were naturally better at adapting to use the boot, or say technology, if we could generalize it. And we wanted to see what kind of traits do these individuals have. So we resorted to flexibility again. We looked at their standing EEG data. That means they're not doing the task yet, but even before that, we are just trying to look at the trait of their brain through flexibility. So we looked at standing EEG and calculated flexibility from there. And it turns out flexibility during standing condition from different brain regions actually predicted adaptation. So here, again, lower muscle use is higher adaptability, and we have a clear positive clear negative correlation. In particular, we observed that the posterior regions were more predictive of how well a person could adapt to the use of exoskeleton boot. There, we also studied some clinical data. So there are, of course, clinical implication as well. Particularly, this was epilepsy. And I'm sure most of you are familiar with this. Epilepsy is one of the most common neurological disorders. It is identified as these spontaneous seizures. Some seizures just emerge focally within one hemisphere. Some seizures are more generalized. They spread to a larger brain area. And modeling epilepsy or this data through network perspective kind of has been really helpful. So we were looking at this problem of heterogeneity of seizures. So this data is ECOG, where electrodes are implanted surgically on the scalp within on the cortex, like inside the scalp. And these are two different seizures from single individual on different days. So in the left one, there's a seizure that emerges, it stays local, and then it dies down. In the right one, a seizure emerges, it kind of starts spreading and then stays for long. So this is a much severe kind of seizure. So focal seizure that remains focal, focal seizure that becomes bilateral. And we wanted to see if there is some role of network reconfigurations in differentiating these two seizures preemptively, so to be able to give the clinicians enough time for some intervention. So we started with similar approach. We used ECOG data, we extracted different connectivities across different time windows. And then we looked at the evolution of network properties. I'm just showing modularity here. So this is directly not related to flexibility, but it turns out that modularity increases in case of seizures that become bilateral or that become more severe. And we hypothesize that this happens. Okay, yeah, I just forgot to mention that this is where the seizure starts. So the change of modularity actually happens much before the seizure actually starts. And we hypothesize that brain becomes more modular or fragmented. And in trying to compensate for that, overcompensation leads to more synchronized activity and generalized seizures. All right, so to kind of sum it up so far, we saw that flexibility across scales and modality captures unique brain abilities. We looked at EEG data, fMRI data, ECOG data across, so it kind of changed from millisecond to days, different spatial scales, and imaging modality, as I mentioned. Another question is, okay, we know that flexibility is good for us, or it at least helps with some sort of cognitive processes, but can we harness it or modify it? So one of the approaches to potentially enhance it or kind of modulate it is through non-invasive stimulation. It could be, stimulations can be different types. It could be magnetic, electric. We particularly use this data which had transcranial magnetic stimulation, repetitive transcranial magnetic stimulation to cerebellum. I remember cerebellum also came up in one of the talks earlier. Anyway, so if we, this cerebellum is this small region at the back of our brain, like closer to neck. And if we just compare it to the cerebral cortex, it has 4 times the more neuron, but it has more homogeneous connectivity. And while cerebral cortex is considered the driver of cognitive abilities, this is kind of considered a modulator or kind of it's believed to have an assistive role in cognitive processing. So stimulating this region is kind of great because it doesn't mess up directly with whatever is going on in cerebral cortex, but it can have, it can push or moderate the activity. So we had this Porta and measure kind of experiment where we calculated recorded fMRI activity before stimulation. There was a stimulation, and then we recorded the activity after stimulation, and we estimated flexibility in both conditions and compared it. And turns out that a significant amount of brain regions, both within left and right hemispheres and within cerebellum, increased in their flexibility. And overall, in general, also there was a higher flexibility present in the brain after cerebellum stimulation. But it might not be true for stimulating anywhere. It turns out that cerebellum nodes have this property of being highly integrative, as in they tend to interact with different nodes that belong to different communities more often than other nodes. And that's why maybe it happens that cerebellum leads to improved flexibility. All right, so to quickly summarize, network flexibility provides a window into brain's ability to adapt to technology, have resilience against environmental fluctuations, resist to external pressure like say sleep changes, and it possibly can compensate for internal malfunctions as well. And it's a powerful tool to apply across multiple modalities and experiments. But just before I end, I want to say quickly that brain flexibility is a multifaceted, or it's a complex concept. There are different ways of looking at it. So one way was to look at these functional networks. There could be another way to look at structural constraints, because structural connectivity is the first basis at which all the interactions happened. There's also a framework inspired by critical brain theory. I've worked on these two a little bit. And then recently I'm diving a little bit into combining deep neural networks with neuroimaging data to get to more explainable kind of AI, and then learn from it where the variability lies. So in terms of modeling the structural connectivity, what we did was we started with white matter connectivity, or the physical wiring of the brain, and combined it with coupled nonlinear oscillator models, which were coupled using this connectivity model. They were delayed, so they generated beautiful dynamics. And then we looked at how computationally stimulating each brain region across different individuals, what kind of patterns of activity it produced. And it turns out that some regions or some functional systems within the brain are designed to be less flexible and more consistent within the brain and also across individuals. And there are some other regions which are more sensory and related to learning and executive function. They are more variable and more flexible, and probably these are the ones that kind of give rise to individual variability in behavior as well. I can skip this. Okay, so the other framework is inspired by critical brain theory, which some of you might be familiar with. It kind of goes long back, which posits that brain is poised close to a critical state, which separates order and disorder, and it was established using avalanches in the brain, which are cascading activity of neural elements, and those elements were found to follow a power law or scale-free distribution. And critical brain theory supports, theoretically, it provides some advantage. It provides dynamical diversity, efficient information transfer, and such. What we observed in this, in this study a few years ago, is that if we looked at avalanches in the brain, macroscopic avalanches in the brain, measured using EEG across different tasks, and tasks here were compared to resting state, passive viewing of images and active viewing of images, where people were trying to control their emotion. The way, even though there was a power law at a larger global scale, the way different avalanches distributed varied between tasks. There was kind of a reconfiguration. And individualistically, how each individual changed in their avalanche distribution kind of guided how well they were able to control their emotions. So this also sort of relates to that idea of reconfigurations in the brain and then its cognitive implication. And this is, I'll mention it very briefly, this is where we are combining some deep learning models with fMRI data to identify different cognitive states. And then we are trying to study where the variability lies in terms of identifying different drivers of cognitive state changes. All right, so I would end with thanking all the experimental collaborators whose data I discussed today, and postdoctoral fellows and students who actually did all of this work. And I would like to thank US Army, DEFCOM, Army Research Laboratory for research funding, and thank you all for your time. Yeah, I think I can end now.
Thank you, Kanika. Thanks, Kanika. Yeah, you cannot see us, but I think you can hear us, right?
Yes, so, I can hear you.
I think even now she's not able to see us, right?
Yeah.
I'm sorry.
That's okay.
Do we have questions?
Oh, now I can see you.
Wait a minute. We have questions. I don't know. If you closed your presentation, can you see something? Maybe?
I can see you now.
Oh, good.
Are there drugs that increase brain flexibility?
That's a really good question. So I would say there might be. but I have not done an explicit study with that. We are planning, and of course, there's a challenge of like drugging human participants. We are planning an experiment with zebrafish, and there we could use different drugs, anesthesia related, and also more recreational kind of things to see how they respond to the brain.
More questions?
Thank you, Kanika, for the presentation. I would like to know if there are conditions in which you have too much flexibility. Is something like that possible? I don't know.
Yeah, I think that's a good question as well. And in fact, we keep thinking about flexibility can't be really good all the time. So maybe too much flexibility could lead to more, attention shift. That might happen in kids with, say, ADHD, right? But I have not looked at ADHD data in particular, but I would like to. So yeah, that could be one situation where flexibility might not be as useful.
Okay, thank you.
Do we have more questions? I have a question, but actually it's not a question. Maybe it's just to check if I understood properly. You show us some data from people with epilepsy, right?
Yeah.
And then,
Yeah.
if I understood right, you can kind of predict when this seizure will, in some sense, spread. Is it all right?
Yeah.
And
Yes.
then you show us something that it's maybe able to, in some sense, stop this increasing of modularity. Is it okay? So my question is not a question, it's maybe just to be sure that I understood properly, but in some sense, you are given some, not solution, but maybe a way of stopping this with some TMS improvement or something like that. Is it all right?
Yeah. So I would say that TMS or brain stimulation exists as a treatment strategy for different diseases like epilepsy, depression, and everything. But the problem is it varies vastly. The outcome varies vastly. It's not effective all the time. So that's one problem. So we need to do more modeling and more work to kind of understand individual variability. And the other thing is, in this particular example, these patients, so we were able to predict, say, 15 minutes ahead of the seizure onset, whether this seizure is going to spread or stay localized. And we don't really know so we propose that maybe there are these kind of network changes that might be happening that lead to the spread of the seizure. But we don't really know at this point how exactly to control for those changes. So, right, like what kind of alterations to give externally that could revert the network back to its like baseline modularity situation. So there's a lot of work that can be done in that direction.
Well, but it still is a good news, right? You
Yeah,
are finding a trigger, so.
Yeah, I think that's promising. The direction is promising.
We still have questions.
Hi, Kanika. Thank you so much for your very nice talk. I was wondering if you probably tried to estimate how much of this flexibility spans in a given task. For instance, when you are learning a sensory motor task, how much it spans to other brain regions, and can you give a specific load for the distance within those that will, I mean, how the distance would contribute to this flexibility account? I don't know if I made myself.
So, if I understood it correctly, are you trying to say that how much of like distributed brain regions given to a reference brain region flexibility would change?
Yes.
If
you can give a specific load for how far is the the node that you're considering on the unit. Yeah, thank you.
Yeah, I think so. Maybe that question is kind of more relevant when we are trying to stimulate a particular region and then see how much of repercussions can happen. So what we found in our data analysis, which I haven't shown here, is that it depends, it varies individualistically, first of all. Some people have like naturally more baseline flexibility and then some other. So maybe it's easier to increase the flexibility of people who are not that flexible to begin with, but if people are like already very flexible, then it's kind of difficult to alter that. And it also depends on where the stimulation happens. So the centrality sort of node within the brain network, which also can change individually. And the third aspect is stimulation protocol. So there could be, for the data that I showed you, there was a 20-minute stimulation, repetitive pulses of stimulation. There are many different ways of performing that stimulation. So it kind of varies with all of that. So there is no clear answer, and that's why probably there is a lot of variability in outcomes of this particular strategy as treatments as well.
Okay, thank you.
We still have questions? No? So let's thank Kanika again.
Thank you.
We have a quick coffee break now.